So what do you guys think of that chapter? It's one of those you definitely got to read more than once, kind of thing. Okay, but so just so you know, too, the exams they're not comprehensive. They tend to build on each other naturally, but like when you get to the next exam or the final exam, I'm not going back and asking you about what's the threshold value for depolarization and that kind of stuff. But you may need to understand principles of, of how action potentials and myelin affect, neurons with somebody with MS, and that kind of thing. So you have those things I'll build, but I'm not going back looking for nitty gritty details. So the final exam does incorporate lots of functional systems, and these things I'll build to that, so just kind of naturally. All right, we're talking sensory system today. We're talking specifically somatosensation. So we're not talking special senses like vision and vestibular and taste and all that stuff. We're just talking sensation of the body, okay? We can grossly divide types of sensation into two broad categories. Superficial sensation, that sensation you feel on your skin, essentially, on the outside of your skin, which can be things like pain, temperature, superficial touch and vibration, light stuff on the surface of your skin. Then we have what's called either, some people call it musculoskeletal sensation or deep sensation, okay? You can have a deeper pain, like a deeper aching type of pain that's deep inside a muscle, tendon, bone. Proprioception in the joint, in the ligaments, in the joint capsule, okay? Those are things like stretch, tension, position, deep pressure, deep vibration, okay, that give you this deeper sense of sensation. How do you feel? We'll talk about, in general, how the process of somatosensation occurs, then give a specific example before we start getting into all kinds of other stuff. So, in general, there's some type of stimulus, hot, cold, pain, stretch, vibration, that stimulates a specific receptor, okay, in your skin, in your bone, in your muscle. It stimulates a specific receptor that may be specialized for that sensation or may not be that specialized in the case of a free nerve ending. That goes up a peripheral nerve out in your peripheral nervous system, right? In one of your peripheral nerves up and through your nerve root, okay? Once it goes into your spinal cord, it enters into a central nervous system pathway that carries that information to the appropriate location, that final destination. Could be in your brainstem, could be in your cerebrum. Those are the two biggest areas of locations we're going to be final destinations for this sensation. A specific example is like a pinprick on your arm that causes a little bit of pain, and that would stimulate, in that case, a free nerve ending is what's typically stimulated when you have pain. That would go up a very specific type of axon in the peripheral nervous system, a certain classification of an axon called an A delta, which is fast pain fiber. Okay, it's like a medium myelinated axon within one of those peripheral nerves. It's going to enter into the central nervous system pathway. It's going to go up what's called the anterolateral system, more specifically the spinal thalamic tract within the anterolateral system. It's going to eventually synapse in the thalamus, the ventroposterolateral nucleus of the thalamus, and then it's going to finally go up to the cerebrum and the postcentral gyrus in a little location called the homunculus that's specific to wherever that location was on your body that is represented on that particular part of your brain, okay? So that's the process, and we're going to walk you through each of those things and start in the periphery and work up to the central nervous system in detail. So I gave you a packet of information that just is helpful kind of things for you to utilize. So this is kind of our guidebook for today and for the next few days of all the different things we're going to talk about. And so you starting on the left there, you've got somatosensation, the somatosensory system, which includes peripheral receptors, peripheral sensory neurons that are in the periphery, the axons, and then the sensory pathways that are in the central nervous system. So this kind of breaks it all down for you of all the different things that we're going to talk about. You'll see we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 pathways to talk about and learn. So in your previous anatomy physiology classes, you might have talked about 2 of those. like spinal thalamic and maybe dorsal column medial meniscal. So this is graduate school, so you get to talk about 11 of them. But the nice part is a lot of these pathways, spinocerebellar, fair number of the spinothalamic anterolateral system, they all start in very similar locations. There may be the first couple neurons in their pathways are the same. It's just where they end up terminating that's a little bit different. So you can group some of these, and you don't have to memorize each one exactly if you know that, hey, these three all start in the same location. They all go up this part of the pathway together. It's just that this one stops here, this one stops here, this one stops here. Okay, so don't let it overwhelm you. Okay, so that's what we'll try to help package it that way. Okay, so that's your guidebook. You also see some other resources in there that includes like some tables, so you can like for your own self-study guides to fill in those tables, some little drawing things if you want to draw things out. These are all on Isidore as well. There's a little thing that says like sensory, somatosensation study guides or something like that, so you can print these off. You can type them up on yourself and those kind of things if you want to. So those should be a little helpful for people. So we're going to start in the periphery and then start working towards where this sensation goes. This is afferent information. It's coming into our body, coming into the central nervous system to be processed. So this is afferent system. So sensory receptors, we can just classify them grossly as mechanoreceptors, chemoreceptors, or thermoreceptors. Okay, mechanoreceptors, they respond to some type of manipulation, stretch, pressure, touch, pull them, push them, whatever. That's what's going to cause them to stimulate and send their signal up the axon, cause them. So those are going to be what kind of protein channels are going to be inside those receptors of mechanoreceptors. Modality gated. Okay, so it's a modality of stretch or so some type of receptors that when you stretch on them, push on them, press on them, they open up, and that's what causes sodium to come in to generate the action potential. Okay. Chemoreceptors, these respond to substances released by cells nearby, including damaged cells following injury, infection. So chemoreceptors, or like we're not talking about specifically like olfaction, but your olfactory system, smell is a chemoreceptors that do that. But in the periphery, we're usually talking about neurons that respond to like histamine and other irritants and those kind of things that cause inflammatory responses and so forth. So chemoreceptors, those chemoreceptors have ligand-gated sodium channels on them because the ligand is the whatever chemical that falls on that particular receptor. Okay. And then thermoreceptors. Thermoreceptors respond to heating or cooling, okay, hot, cold. And those that have modality-gated channels. We can also divide, whether it be a mechanical, chemical, or thermoreceptor, we can grossly divide and put all receptors under one of these two classifications of either it's a tonic receptor or a phasic receptor. Some receptors, irregardless of the type, are more tonic in nature, that they respond as long as the stimulus is maintained. You push on it, you press on it, there's a stimulus response, but as long as you maintain that pressure, it keeps sending a signal. It keeps generating action potentials to let you know that that pressure is still there, or that stimulus is still there. Some receptors are much more phasic in nature. You push on it, it generates an initial action potential, but then it stops. Even if you keep your finger on it or keep pushing on it, it stops generating action potentials, or it does it at a much lower rate. Okay, and then when you let go or take your pressure off that receptor, it generates another action potential, but then it quickly stops generating action potentials. Okay, so those are phasic receptors, and they're both helpful. When you first sit down in a chair or something like that, boom, you stimulate a lot of phasic receptors that tell you, hey, I just landed on this chair, there's pressure here, there's pressure there. Some of the phasic receptors, they then pretty quickly say, it's not important I don't need a lot of sensory information about this now that we're sitting and we're stable and we don't need to worry about our posture and our balance too much. And so those turn down, but you still have some tonic receptors, because if you actually think about it, if you consciously think about it, you can feel the pressure still on your butt and your back and those kind of things. And you also, over time, if you stay in one position too long, Some of those receptors will actually start to cause pain, and that's why you just shift automatically. And those are some tonic free nerve endings that are continuing to monitor how much pressure is being applied in the background and stay turned on so that it knows to shift my weight now before I cause damage to my skin, those kind of things. So phasic receptors are really important for during dynamic motion and dynamic things that are happening. Tonic receptors are more during when you're in static positions and static postures, or some stimulus that is, especially noxious stimuli. Your free nerve endings that are pain receptors tend to be more tonic in nature, because you want to know that there's pain going on. When you put your hand on a stove and it's really hot, you don't want just an initial like, ooh, that was hot, and then your receptor to turn off, and then your hand starts smoking because you no longer have. You want that pain the whole time that's on. That's going to be sending a signal, pain, pain, pain. So those are more tonic in nature. So here's here's the stimulus right here. So you apply a stimulus, and this is the strength of the stimulus. It's relative, whatever, but here's a stimulus that you apply to the skin. Let's say it's pressure. You push on somebody's skin. If it falls on a slow adapting or tonic, so they call it slow adapting or tonic, you get an initial burst of activity or an action potential, or more, multiple action potentials that stimulate it, and then it falls off a little bit, but then it maintains a certain level of activity as long as the stimulus is maintained. Okay, so the stimulus is being maintained at the same level, but you get an initial little burst, but then it falls off, but it's still greater than zero. It's still above that base point. And then once you take the stimulus off, it falls back to its baseline. Where a rapidly adapting receptor, okay, or a phasic receptor, the stimulus comes on, you get this big burst, initial burst, but then very quickly it stops, and no longer is it responding, even though the stimulus is still being maintained there. And then when you take the stimulus off, you can get a little off response, where actually in that case it hyperpolarizes, and it's a little bit hyperpolarized in that case. Any questions on we're just talking general classifications of receptors right now. We'll get into more specific receptors in a few minutes. Once the receptor has been stimulated, it's going to send some action potentials up a axon, okay, in a nerve. So it might be in your ulnar nerve, or your, nerve in your leg, the perineal nerve, or something like that. So you might be sending a signal up one of those nerves in these little bundles of axons, right? And those axons are classified as certain types of axons based particularly on how fast their diameter is the main thing that classifies these, okay? And the diameter often is dependent also too on how much myelin they have on them, because myelin accounts into the diameter of the axon, okay? These are afferent nerve fibers, axons. The cell bodies, for the most part, are located outside of the spinal cord. Okay, so what's unique about these is the cell body is outside the spinal cord in that dorsal root ganglion. Remember, there's a little ganglion that contains the cell bodies. It's that pseudo-unipolar cell that, it's got the cell body and has a little stem that comes off, and it branches out into a peripheral and a central axon. These are those peripheral neurons, okay, that are going down into your, going to your different parts of your musculoskeletal and skin, okay, for sensory. Or if it's a cranial nerve, like we're talking about facial sensation, like or proprioception to our jaw and those kind of things. Those are also somatosensory nerves, but they have a cranial nerve ganglion. So that's the equivalent of the dorsal root ganglion in the spinal cord. Like you have the trigeminal ganglion. Our big nerve that supplies our face sensation is the trigeminal nerve. And it's got a trigeminal ganglion. That's the equivalent of the dorsal root ganglion in your spinal cord for afferent information coming in. So these are the types of classifications of axons, and they're by diameter. The larger the diameter and the more myelinated, the faster it's going to be. So on the bottom, this is the maximum speed in milliseconds that the action potential can travel on that nerve. Okay, so starting up at the top here, you have C, classified as C axons. Okay, those are only 2 meters per second, 2 meters per second, which is still pretty fast. I mean, that's 2 meters is from my toes to my head. I'm 6 foot, in a second. 2 meters per second. So it only takes still a second to get there. A deltas, those are called A deltas. Those are 45 meters per second. Type 2, and this should be not AB, this should be an A beta. Okay, so it should be an A beta like that. But so type 2 or A beta fibers, those have about a 90 meter per second speed. The 1B fibers, that is a B, 1B, those are 120 meters per second, and the 1As are 130 meters per second. Okay. And you're going to see when the 1As and 1Bs they're really important, especially for the rapid proprioception associated with joints and muscle spindles, where you have to make really quick automatic adjustments to your posture to keep yourself from falling down, provide your quick stretch reflex responses and so forth. Those are involved with that because those have to happen really rapidly. Okay. So here's your peripheral sensor out here. So in this case, that looks like it might be a Pacinian corpuscle or Merkel's disc or something like that, but it's a specialized receptor. Okay, that would be actually a mechanoreceptor. If it has a really specialized ending on it, it often is typically going to be like a mechanoreceptor of some type that's specific to some type of stretch or pressure. But here's the peripheral axon, right? Here's that cell body lying in the dorsal root ganglion. And remember, there's lots of them in here. There's like more than, and then there's lots of axons that are coming out to make up that peripheral. Going to go into that peripheral nerve. This actually be the nerve root at this point, and so you have multiple axons coming in to this dorsal root. This is the back part of the spinal cord, associated with those cell bodies of those pseudo-unipolar long axons. They can be long or they can be short, depending on where they're going in your body. If it's coming from your lumbar spine going to your toe, that's a long axon that's going to go all the way down to your toe to do that. And it's these axons, both here and here, that are classified, as the 1As and 1Bs and those kind of things, is depending on how myelinated and what the diameter of it is and what its function is subserving. Okay. Let's focus on superficial sensation a little bit, okay, and then we'll talk about deep sensation. So this is just stuff more on the dermis and epidermis of your skin. It was primarily what we're talking about here with superficial sensation. Let's talk about the concept of what's called receptive fields on your skin. It's the area of skin innervated by one neuron, okay, so one single neuron. So you can see this area in yellow. This is all going to one neuron. So this is going to go up. Eventually, it's going to go into a dorsal root ganglion into your spinal cord, okay? But that's the area of skin. It branches, and this may be free nerve endings. It may be Pacinian corpuscles. Who knows? It may be petrichal ending. But anyway, it's going to subserve a certain region of skin, okay? A certain region of skin. Same thing with the one here nearby. This blue one is just showing you another one nearby that's supplying or receiving information from that area of skin. That's the receptive field, okay? This is showing you if you're doing that 2-point discrimination is a way of seeing how sensitive you are and how your ability to discriminate between whether you're feeling 1 or 2 points when somebody touches you. Did we talk about that at all I don't know if I did that when we did that real quick little session on sensation. We may not have talked about 2-point discrimination. So one of the things, there's this little wheel, and I'll show it to you when we do our little quick lab thing. It's got different little points on it, and they're different distances. It's a little, graded point thing where it's got one that's like 6 millimeters, 7 millimeters, 8 millimeters, 9 millimeters apart, these two little points. And then it has one where it's just one point coming off kind of by itself. And what you do is if you suspect somebody's having some sensory problems in an area, typically you're doing this to the fingers. It's really the only place you typically, but you could do it anywhere in the body, is you would touch. And you would say, I'm going to be touching you with this little wheel. I want you to tell me if you feel 1 point or 2 points when I touch you. And what you do is you kind of alternate by kind of, making sure that they don't predict when they're going to get to the feeling 2 points. But so you may touch them sometimes with just 1 point, but then you touch them sometimes with the 2 points. And you ask them, just tell me 1 or 2. So as you're doing this, if you're touching them with 2 points and they say 2, that's good. If you touch them with 2 points and they say 1, then that could be a problem. But it depends on where you're touching them. If you're touching them at the very tips of their fingers, okay, especially like the thumb, index finger, and middle finger, you should be able to feel probably about 4 to 5 millimeter difference, okay, and experience that as sensing 2 points. Because what's happening is in your tips of your fingers, your lips, even some of the tips of your toes, you have very small receptive fields. You have lots of nerves supplying very small regions of that skin in that area. So there's a very good chance, in order to you to sense that there's 2 points, those little points have to fall in 2 separate receptive fields. If you have a really large receptive field and you touch and those 2 points fall within that same receptive field, your body is going to perceive it as just 1 point. Does that make sense? You have to have the points fall in 2 separate receptive fields for your body to say, oh, that actually is 2 points because it's falling in 2 different locations. So the smaller the receptive field, the higher resolution, right, of the information you get, because you have more individual nerves supplying smaller regions of skin. So when you're trying to manipulate an object, I can feel every little texture and ridge on this because I have all these little tiny receptive fields that are providing this very discrete information about very small parts of this object. Does that make sense? And they're going to all these locations in my brain that tell me about the three-dimensionality, the texture of this object because it's falling on lots of little receptive fields. Or if you go to like my back here, the receptive fields are massive. There's only one nerve that's going out to supply a really large area of skin. So if I do a 2-point discrimination, it's going to take like a point this far apart for me to say that, oh, that's 2 points. The key though is you got to like touch the 2 points at the exact same moment. If you go dink dink like that, then that gives it away. Even if it falls in the same field, you can feel it 2 points hit at separate times. You got to hit them right at the same time. You'll still perceive it as just being one. You'll perceive it as being 1 point if it falls into that one large receptive field. You have to hit 2 separate receptive fields. Now you might get lucky on a back and happen to just get the 2 right where the 2 receptive fields. There's usually some overlap. So you see there's overlap here. So, but so you have all these other receptive fields that are all overlapping out here. So as long as 2 points fall in 2 different receptive fields, you're going to be able to perceive it as 2 points. Okay. Nope, they would just perceive it as 1, yeah, in that overlap region. Yeah, but that's a good point. They would just feel the 1 point, even though it's hitting, because it would happen the exact same moment. You'd have to get in those 2 fully separate receptive fields for that. You could be this way though. I see what you're saying, but you could, if you had 2 points, if the 2 points were here. So you had 1 here and 1 here, you could probably still feel that as 2 points because you still hit that receptive field overlap. So the more overlap you have, just the better chance you're going to feel 2 points no matter how close they get together. So I'm not sure why they have this guy diving, but anyway. This is just showing you the relative receptor field size or the 2-point discriminatory sensitivity of different regions of your body. So 42 millimeters, you have to use something that's 42 millimeters wide on the back for most people to say, hey, you're touching me with 2 points at the same time, or you're hitting me with 2 points. Anything narrower than that, you're going to perceive it as just 1 point. It's when somebody touches you on the back with that. It's just, the hard part, when you get that wide, it's hard to hit them both at the same time without somebody recognizing you're hitting with 2 points. You have to be super careful when you do that because you also have your back is curved and it's hard to get them both to hit at the exact same time when they're 42 millimeters apart. Look at the calf. The back of your calf has very large receptive fields. It's got even larger than your back there. I don't know, it's like 50 or so millimeters wide on the back of your calf because you don't use your calf or your back to manipulate anything. All you need is gross sensation. Is there pressure back there when I sit? You don't need to manipulate objects and sense textures or anything like that. So you don't need a lot of density receptive fields. So you don't want lots of extra information coming from those areas and sending them to your brain, taking up valuable real estate and energy in your brain. You only want them to come from the parts that are important, like your fingertips, especially your mouth and tongue. Those are the ones that, and when we look at a homunculus, you'll see that most of your homunculus is hands and face and mouth and tongue, okay? Your feet, they take a little bit, but even the big toe, you see the big toe is still pretty wide. Oh, wrong screen I'm drawing on. And then index finger and thumb are really kind of, that's the ones you use the most, right? They're the ones that are have the smallest 2-point or the finest 2-point discriminatory ability, okay? Lip is close there too. Still on superficial sensations here. So these are the skin sensations. There's different terms used to describe things you feel on your skin. There's what's called fine touch or discriminative touch, okay? This is touch that when somebody touches you or you feel a touch, you can say, yep, I feel a touch and I felt it right there. And you can point to the exact location on your arm, on your leg, wherever that somebody touched you. That's called fine touch, okay? Where somebody touches you, you could like say, if you did it to a patient, you might say, I'm going to touch you lightly with this cotton ball. And tell me, first of all, if you can feel it. And then I'm going to have you open your eyes, and you're going to point to exactly where I touched you. So that's a different level. And we should have showed you to say yes, yes when you feel it. That's coarse touch. That's just saying, yeah, do you just feel it? But if I say, close your eyes, I'm going to touch your arm, I touch it, you go, yes, and then I say, open your eyes, point to where exactly I touched you. That's fine touch. Localized, discriminative touch is fine touch, okay? And those are using specialized receptors, okay? Meissner's, Pacinian, Ruffini's, Merkel's discs, and the hair follicle receptors. If you stimulate a hair follicle, that's using these are all mechanoreceptors that have a specialized nature to them. That's what provides you this sense of fine touch and localized touch. Okay, and it's got a little more, fidelity to it, a little more characteristic to it. You can sense the pressure and which way that touch is moving on your skin and those kind of things. That's what these things provide, that high fidelity discriminative touch. Then there's coarse touch, which is poorly localized. This is where you touch somebody and you say, did you feel me touch you? And they say yes, and you say, point toward me where I touch you. Like, well, I know it was down here in my arm somewhere, but they can't say it was right there. Like, yeah, you touched my left arm, maybe somewhere in my forearm region, but I can't exactly tell you where I touched you. That's just coarse touch. It's also a part of the what's called itch and tickle touch. So that itch and like when you get a really bad itch, sometimes you can't find exactly. It's like I know it's coming from here, and it seems like it spreads sometimes, and you're like, it's hard to localize exactly where the itch is. You just know it's in this general region. So itch and tickle pathway is actually carried. Itch and tickle is the precursor to pain. So itch and tickle is carried on the same pathway and stimulated by the same receptors, which are free nerve endings that you just haven't stimulated them enough to cause pain. It's before they turn into pain is what itch and tickle is. Okay, you're stimulating, you're waking up those free nerve endings, and they feel it as sort of an irritation, but it's not a frank pain yet. Okay, but that also can help you with this coarse touch is carried on these pathways that are actually different than the fine touch pathways. Coarse touch is carried on actually pain and temperature pathways, but it's itch and tickle. Okay, coarse touch is using just free nerve endings, which are non-specialized. Free nerve endings are non-specialized receptors, so they just provide you this very gross form of touch, but not this very nice discriminative touch that we just talked about before. Pain. So pain is free nerve endings, just like the coarse touch is, but just when you reach a certain level of intensity of a threshold of stimulus on that free nerve ending, then it can cause pain. Okay, and when it's acting as a pain receptor, that's called a nociceptor. Okay. These free nerve endings, what's interesting about free nerve endings, they're kind of cool. They can be stimulated by mechanical means, they can be stimulated by thermo means, or chemical means. So the free nerve endings are the do-it-alls, but they provide not great fidelity of information. They provide the itch and tickle, and they provide pain, okay? But they don't provide pain until you you mess with them enough. You have to like really push on it for a lot. You like push really hard on that free nerve ending. You've also stimulated some of these other Pacinian corpuscles and Meissner's is saying somebody's pushing on me right there, but it's the free nerve endings that you push on them long enough, ow, that starts to hurt. That's causing pain now, okay? Or you are feeling warmth as you put your hand on the stove, and as it gets hotter and hotter, all of a sudden you cross that threshold, now it's pain because you need to have that protective response. But you're stimulating free nerve endings that are acting as thermoreceptors in that case, okay? Or if you have damage, you cut yourself and cells release a lot of histamine or something in the area, bradykinin, and they cause inflammation of free nerve endings around that area and cause this pain signal because they're acting as a chemoreceptor in that case. These free nerve endings can act as a mechanical receptor. As long as you stimulate them to a certain level, they can cause pain. Temperature are also free nerve endings when they're not if you don't stimulate them to the level of pain. Okay. This is showing, yeah.
When you stimulate those neurons, do they get more localized when you get to the point of pain? Because like if you prick somebody's finger,
yeah,
where it causes pain, that seems pretty localized. But you said the free nerve endings are general.
So they're general in their function. They can kind of do a lot of things. When you do stimulate them to the level of pain, they can cause pain. And it's actually when you do have pain, though, it can be very localized initially. But then if pain is maintained, it gets more diffuse and it gets more deep aching pain, then it's harder to tell where it's coming from. Okay, so it's different than under the coarse touch conditions. When you're not stimulating it to that high threshold, it's very hard to localize. But if you do prick yourself with a pin to the point of pain, then you can localize it a little better. Does that make sense? So this is just a cross section through some epidermis and dermis right here, for superficial sensation. So in hairless skin versus hairy skin, there's some slight differences. So with hairy skin, you can see there's certain things that tend to be in the hairy skin. So you have this is the petrichial endings. These are these little nerves that wrap around the hair follicles. That's a petrichial ending, or a hair follicle nerve ending, or peritracheal nerve ending. That peritracheal wraps around the hair follicle. You do have more Merkel's discs. These Merkel's discs sit right on the surface of the skin, or right that junction between the epidermis and the dermis in hairy skin. You have more Merkel's discs. Doesn't mean you don't have any Merkel's discs in hairless skin. This is just kind of showing you that, accentuating the differences between the two. It's just saying you tend to have more in hairy skin of Ruffini endings and Merkel's discs and the hair. And then in hairless skin, you tend to have more Meissner corpuscles, okay, and more free nerve endings. However, you still have free nerve endings in hairy skin. You still have some Meissner corpuscles in hairy skin. It's just showing you there's a little bit greater concentration of slightly different types of mechanoreceptors and free nerve endings in different hairy versus non-hairy skin. Okay. This is a very good table to know. This one kind of summarizes about receptors, okay, for peripheral nerve receptors. So on the left, you have the receptor. So here's free nerve endings. And it tells you about the anatomical characteristics of it. So you can see that free nerve endings are minimally specialized. They don't have a lot of specialized look to them. What are the associated axons? When you stimulate a free nerve ending, it's going to get sent up what type of a peripheral axon? It's going to either be a C or an A delta. That's the classification of axons. So those tend to be, the C's are slow, A deltas are a little faster. They're called fast pain, but they're not that fast compared to some of the other, like 1A's and 1B's and those kind of things. Remember, that's like 2 meters per second and 45 meters per second. Free nerve endings are located in all skin, okay? Like, even though that picture showed free nerve endings not in hairy skin, it's in all skin. And this is its function. What does it tend to do? Pain, temperature, and crude touch. That's what free nerve endings are mainly, the type of information they're transmitting or functioning to show. What is its rate of adaptation? These are slowly adapting, right? Pain receptors are slowly adapting. As long as there's something causing us pain, we want them turned on and keep telling us to get rid, because we want to minimize that pain. what is the level of activation on this? It's pretty high. You have to really push on it, heat it up to get it to actually cause pain and send a signal. So there's a pretty high threshold for activation on that. So that's the free nerve endings. And then we work our way down. I'm not going to go through all of them, but let's just do one more example. Let's just do Meissner's here. So Meissner corpuscle, what's its anatomical characteristic? It's an encapsulated, specialized structure, and it's between the dermal papillae. Don't worry about that, but it's just close to the dermis there. It's that one that's right there between the dermis and epidermis. It sends its signal or action potentials on axons that are A beta axons, okay? It's principally in glabrous skin, which would be the, this is a Meissner's corpuscle. Sorry, Meissner corpuscle is here. I was saying Merkel's discs. I'm sorry, back there. So Meissner's corpuscle is right here between that layer of that dermal layer right there. But it's glabrous skin is hairless skin. Okay, that's all that glabrous skin means, is more hairless skin. It responds to touch. It's and pressure. It's very dynamic. As you push it on and off, it can really sense. It senses nicely. It's sensitive to changes in pressure and touch. And it's rapidly adapting, right? And that's so it's good at sensing changes in pressure because it's rapidly adapting. It turns on and off very quickly in response to changes in touch. And the threshold for activation is low. It's very low threshold because it's a touch. That gives us a sense of proprioception or texture and touch on our lips, on our fingers, those kind of things. It's very important for giving us that information. So it's a low threshold of activation. Okay, you want to be able to feel things even with a very slight pressure, and sense that. So here's some things as you're looking at this chart. Again, think about grouping things together. So if you look at all of these are A betas as far as the axons. All of these right here, all I really want you to know about these is these are all specialized types of receptors. Don't have to worry about the details. Just know that free nerve endings are minimally specialized. All the rest have some special anatomical characteristic to them. Okay? Then look at like threshold of activation. The only one that's high is here. All the rest are low activation. So if you just know that free nerve endings are high, all the rest are low level of activation. Rate of adaptation isn't so critical for you to know. A lot of detail on that. And as you start to get to know, when we go through the pathways, you'll start to just associate these with certain types of function. Okay. Then all you have to know here is these are 1As and 2s. These are 1 betas. And then these are the C's and A deltas. So kind of group things like that as you're looking at this table. We're still on superficial sensation here. So superficial sensation contributes to this thing of, right? Shouldn't we have allergies this time of year? Dermatomes. You guys know dermatomes. It's the area of skin innervated by a single spinal nerve. That's the whole nerve, which has lots of axons in it, right? Okay, so it's got lots of axons, and it's supplying, though, that entire region of skin for a specific spinal nerve, okay? The ones that are most clinically relevant for you guys are C5 to C8 and L4 to S2, because those are the most common ones you're going to see people having a problem with. In the clinic setting. When you're working with people with neck problems and back problems, those are the locations where you're going to be able to see the most dysfunction. Between C5 and C8 is the most common areas of disc bulges or degenerative disc disease, stenosis, and those kind of things. That's going to cause like radicular symptoms into their arms or legs, or where you're actually able to test reflexes or sensory areas. Once you get into the trunk, it's a little harder because, intercostal nerves and those kind of things that come around, they may have something like that, but you're not able to test reflexes. There's a lot of overlap of those, so it's harder to really define. And also, they're not involved as much. Okay, and also you get above C5, you're talking about the head, and skull and stuff that has those sensory areas, and it's harder to to screen out. So we're talking about these supply mainly your upper extremities and your lower extremities are the most clinically. So if you really, I really want you to focus if you're learning dermatomes on those dermatomes specifically are the most critical. Um, Peripheral cutaneous nerves in the upper extremity, the key ones there. Once you branch out and start to go into your terminal peripheral cutaneous nerves, the larger ones anyway, before you get to the very terminal things, median, ulnar, radial nerve. Those are the big ones for the upper extremity. For the lower extremity, mainly like femoral nerve and superficial perineal nerve for sensory area. Those are ones that, from a peripheral standpoint, when you have damage, outside of the spinal nerve root, those are the ones that I think are most clinically seen or relevant for you to see. So let's talk about Donna dermatome. I just found this in an old textbook. I just thought it was hilarious that this is how they showed dermatomes in one of these old textbooks. So very appropriate, I guess, here. Very. I don't know how you see all the dermatomes is the problem, though. She's like, oh, like this. So it's like, so that must have been back from like the 50s or something like that. So this is from your book, which I don't like this one either very well. So on one side of the body, you see the dermatomes. The other side is when it actually branches into the peripheral cutaneous nerves distributions, okay? So the ones I was pointing out to you that I think are the most important would be, C5, C6, C7, C8, T1. Those are the ones that are really the most important from the upper extremity. And then for the lower extremity, we're talking L4, L5, S1 are kind of the ones that tend to be most involved when you have somebody has a spinal nerve problem, nerve root, right where it's coming out of the spine. If you have pressure damage to those, could cause dermatonal loss of sensation if the pressure damages enough. Not always. Or they may just have paresthesias. They might just have an abnormal sensation. If it's just pressing slightly on a nerve, a sensory nerve, it causes paresthesias like tingling, that itch and tickle kind of sensation where you don't have enough pressure to cause pain, but it's enough to cause some tingling sensations and so forth. And then when we get out to things like, radial and ulnar nerve and those kind of things, so that's more down, hand and so forth. So where we can see, here's ulnar nerve, and then we've got some, radial nerve here, median nerve down in here. So the kind of general, those are the areas that you're looking at mostly. You could have some axillary nerve damage that causes some just damage to the axillary nerve. You just have to kind of differentiate that from a dermatonal pattern. So, because C5 though continues to go down in a certain region, so you have to kind of explore that. Is this a peripheral cutaneous nerve problem or a dermatonal problem, based on kind of outlining where the loss of sensation is? Okay. Also, that the nature of the injury. If somebody was in an accident and they got their fibula, broken, and they have numbness now right here, that's a superficial perineal nerve injury if I ever saw one. It makes sense that that's not a radicular problem coming from the lumbar spine like S1 or something like that that's causing that problem. Okay, so those are some things that help you understand that. Let's get through musculoskeletal sensation, then we'll take. You know what? Let's take a break now before we get into this. Any questions before we pause? Okay, come back. So we just spent a lot of time talking about we're still on the periphery out here in our sensory system. We talked about superficial sensation, type of receptors, the type of modalities of sensation we perceive in the superficial regions. Now we're going to go a little deeper into what we call musculoskeletal sensation, kind of both literally and figuratively deeper into muscle, ligaments, tendons, those kind of things, how they sense or have sensation or somatosensation. We're going to talk about the muscle spindle first. It is a sensory receptor that's in your muscles. It influences your motor output. And when we get to the motor system, we'll talk about how the ability of the muscle spindle to sense length and tension of your muscle helps influence motor output. But we're just going to talk about it for the moment, kind of focusing on what it does as a sensory organ in your muscles. So the muscle spindle, it's embedded in all skeletal muscle. detects both length of your muscle at any given time and how, if your muscle is changing length, how fast that change is occurring. Okay, those are two things that are very important. It'll statically tell you what's my muscle length in the static position, but it'll also tell me how quickly my muscle is lengthening or how quickly it's shortening. Okay, that's that muscle spindle is a sensory organ that tells us, because it's embedded in the muscle, so it can sense the length of the muscle and the tension in the muscle. So within a muscle spindle, a muscle spindle is fusiform in nature. It's tapered at both ends. That's what we mean by fusiform. So it's a sensory organ that's fusiform and it's a specialized mechanoreceptor. So this is just a mechanoreceptor that's very specialized in nature, and it's fusiform and it's embedded in the muscle. Okay, and so inside of the fusiform structure is these intrafusal fibers. Right, these are the intrafusal fibers. There's these two different types of fibers. One's called the nuclear bag fibers. This is like a clump of nuclei in the middle of this fiber, which we'll show you pictures in a second. Then there's the nuclear chain fiber, which is a straight fiber with lots of cell bodies that are all lined up in a chain. Okay. You have afferent nerve endings. Those are the sensory part of the muscle spindle. Okay. This is what that sends the information from the muscle spindle. It's going to go back into your spinal cord. Okay, the afferent fibers. That's the signals. That's the sensory endings. You have what's called primary and secondary endings. Okay, sensory nerve endings that are in your muscle spindle that are attaching or detecting changes in these intrafusal fibers. The primary fibers are also known as annulospiral nerve endings. The annulospiral nerve endings are associated with 1A axons. The information is carried on a 1A type axon, which is that's the fastest axon you got. Those are that 100 meters per second, 120 meters per second, because the muscle spindle is really important to provide very quick information about what's happening with my muscle so it can get in my spinal cord, get to my brain or my brainstem, make an adjustment, and go back and make on-fly adjustments to my muscle function if it needs to. Okay. The primary has both phasic and tonic qualities to it. Okay. So the primary nerve endings associated with the intrafusal fibers have both phasic and tonic qualities, meaning that you stimulate it through rapid movement. It'll send a stimulus, but then you maintain that new length or position. Then the tonic part will take over, and it'll still send signals about what's going on tonically with this new change and new maintained position of the muscle. Okay. The secondary nerve endings, also known as the flower spray nerve endings, they are carried on type 2 fibers, a little slower than the 1As. They are specifically more tonic in nature. They're telling you mainly about the resting length of the muscle, the new resting length, or the tonic resting length of the muscle, where the primary endings are really important for both the phasic, but they also contribute to the tonic sense as well. But they're the ones that are really for the changes in muscle length. You also have efferent. You have motor fibers going to the ends of this muscle spindle. There's a few little muscle fibers that are inside this fusiform body, okay? And these are called gamma motor neurons that go to the little muscle fibers on the inside of this muscle spindle. There's a few little muscle fibers on each end of this muscle spindle that can help adjust the gain or the tension and adjust the sensitivity of the muscle spindle to stretch within the muscle. Okay, so they're like almost like having a little spring on each end, and they can tighten the spring or loosen the spring of the muscle, the little fibers on the inside of the and these are intrafusal muscle fibers. Your musculoskeletal, your muscle fibers, they're called extrafusal muscle fibers. These are intrafusal muscle fibers. And they have very specific gamma motor neurons that go to them, where your muscles, the muscles outside of the muscle spindle have alpha motor neurons going to them. Okay, they're slightly different in their characteristic and how fast information is transmitted. But we're not focusing on that right now. But they help to maintain the length of the muscle spindle. Okay, so that if I were to here I got a muscle spindle embedded in my muscle, and I contract my muscle my muscle spindle initially starts to shorten, and it sends a signal saying your muscle's shortening. It's happening about this fast, but pretty quickly that muscle spindle goes on slack. And if I assume this new position, if my muscle spindle's on slack, it no longer has very much sensitivity to it. Because if I were to stretch it out again, there would be a delay before it would stretch again and send another signal telling me it's going the other direction because there's kind of slack in it. But if I flex my muscle, the muscle spindle briefly goes on slack and sends a signal saying, hey, your muscle's shortening. Then once I've achieved this new position, those little interfusal muscle fibers tighten it back up again. So now the muscle spindle has a little tension in it. So as soon as I start to extend it again, it senses that stretch again. There's no slack in it. It has to be taken up first, but to send another signal. Okay, setting the sensitivity or the gain of the muscle spindle that detects the tension and rate of length and tension. So you have to be able to adjust the length of that little muscle spindle within the muscle. Because the muscles are contracting, the muscle spindle has to kind of contract with the muscle. Does that make sense? And that's what efferent nerves are going to do. So this is just showing you that fusiform structure, muscle spindle within the within the striated muscle itself. So this right here is a alpha motor neuron going to the actual muscle, okay, to the striated extrafusal muscle fibers, okay, that's an alpha motor neuron. These green guys right here, this little green ones, those are gamma motor neurons. That's what the gamma symbol looks like a Y there. It's a gamma. Those are the gamma motor neurons going to, these are the intrafusal muscle fibers. Right at the ends of here are little interfusal muscle fibers to help adjust the tension within that muscle spindle. Here's the nuclear bag, kind of in the middle there. Nuclear bag fiber right here. It's got like a bag. It's a little bit, big in the middle, kind of like a bag, and got some nuclei that are associated there. Then this also looks like another nuclear bag fiber down here, but this one up here on the top, here's a nuclear chain fiber, and you can see all the little nuclei just kind of lined up in a row here, and it doesn't have that bulging center of it. That's a nuclear chain fiber. Okay. I think I have another picture now that we're just going to simplify and look at one nuclear bag fiber and one nuclear chain fiber. I'm knowing that there's lots of them in there, but we're just going to look at just the nuclear bag and nuclear chain. So this is the nuclear bag on the top, and you can see all the cell bodies all mostly, there's a few out here, but mostly lined up or not lined up, but clumped together in the middle there. This is the annulospiral. See how it spirals around the nuclear bag fiber? Annulospiral, primary nerve ending. 1A afferent. So when that nuclear bag gets stretched, it's going to send a signal on a 1A afferent to say, hey, my muscle is changing length and it's happening about this fast. This is the phasic part of it. The nuclear bags are really good for detecting the rapid changes in length because you have all these little cell bodies and it's clumped up in the middle, so it stretches really good. Like it detects stretch very good. Not stretch, it detects changes in length very well because it's kind of clumped up in the middle. It can allow it to change shape really quickly and cause a phasic response to say my muscle's really rapidly or slowly changing length or whatever, and send it up that 1A fiber. Remember we said the primary nerve endings have both phasic and tonic characteristics. Well, here's a branch of that 1A that's wrapping around the nuclear chain fiber. The nuclear chain fiber, since it's just this long lined up fiber with cell bodies lined up, it doesn't change shape very much, but it's very good at sensing tension. You just kind of pull on it and it just gets a little tighter, but it doesn't change shape like the nuclear bag fiber so much. So it tends to sense tonic sensation in changes in length and tension in the muscle, not the rapid change in length. So once you achieve this new length, it senses the new resting tension in the muscle, whether that's shorter or longer, and then it's tonically telling you all the time this is the new length of the muscle. Does that make sense? So that's why the primary nerve endings they have both phasic and tonic natures to them, and they send that signal on this really fast 1A axon that's going to go into your nerve and into your spinal cord eventually. Okay? Where here's the type 2 secondary nerve endings. This is called a flower spray. Even though it shows it wrapping around a little bit here, a lot of it just kind of sprays out like a flower, flower spray ending. It doesn't wrap around so nicely as the annulospiral fibers, okay? And so this is a secondary ending. This is type 2 axon, a little slower. It's only sensing the tension of the muscle. It's not giving you much information about any changes in length. It's just changes in tension and static, position of the muscle. And these are the efferents. These are the gamma motor neurons going to the little muscle fibers on each end. You'd have them probably going to the other side over here. You'd have them not just a one end, but both ends to kind of adjust the length and tension when you do shorten and lengthen your muscle in that muscle spindle. Okay? Sorta? We'll come back to muscle spindles again when we get to the motor system and then talk about how it influences motor output, through reciprocal inhibition and agonistic, excitation and all that fun stuff. We're still on musculoskeletal sensation. We're still on these very specialized receptors. The next one we're talking about is a Golgi tendon organ, GTO, okay? Here it is. It's not as specialized. These are encapsulated nerve endings in the tendon, so they're kind of threaded, in the tendon here. So this is the muscle here. It's the musculotendinous junction area. So it's sensing, the GTO senses tension in tendons, okay? They're going to send their afferent information about tension up a 1B afferent. Again, still very fast, just a little slower than the 1As. It's as similar as the 1Bs and 2s have similar 90 meters per second, a little slower than the 120 meters per second on the 1A, but they're still pretty darn fast. But they're sensing tension in the tendon, okay? Musculoskeletal sensation still. Now we're talking about joint receptors. So we started muscle, we did tendon, now we're on joint receptors, okay? So we have sensation within our joints themselves. These respond to mechanical deformation of capsule and ligaments. So your joint capsule and your ligaments, okay, which are surrounding your joints. You'll see some familiar names here because probably the same person that found it in the surface of their skin found it in ones in the joints that look very similar. So we have Ruffini's endings, which are very similar to the Ruffini's in the skin, but these are down in the joints, okay? We also have Pacinian endings. These are very similar to Pacinian corpuscles that are in the skin, but these are down in the joints, okay? We have ligament receptors and we have free nerve endings. You can hear them referred to in that way, or some people will just say type 1, type 2, type 3, type 4 joint receptors. I think it's more common to hear like type 1, type 2, type 3 when you're reading research articles or something like that. You're probably more likely to hear the type receptor with its Roman numeral versus the, name of it, okay? You'll see that they're all pretty fast. They're either using the axons they carry that information on are either type 2 or 1Bs. The only one that's slower is there are free nerve endings that carry it on A, delta, and C. That's for inflammation and pain within your joints. Those are the pain receptors within your joints. The first three are all mechanoreceptors providing position sense, okay? Ruffini's endings, or type 1s, tend to get their most stimulated at the extremes of motion. When you get to the very ends of your joint motion, they take a lot of stretch. They're a little higher threshold. Even though they're still low threshold, they're a little higher threshold than the other ones. They tend to require a little greater stretch. Pacinian endings tend to be most active during dynamic as you're moving the joint, not in a new. Once you settle into a new position, it's primarily like Ruffini endings that are going to give you that information. Pacinian endings are very good at giving you dynamic joint motion sense. And then the ligament receptors are similar to the GTO, but in the ligaments. So changing tension in the ligaments around that joint are going to help with that. The other ones are mainly in the joint capsule, okay? The Ruffini endings and Pacinian endings. Okay. Just showing you some general locations of this is a knee joint, showing you where some ligament receptors, some free nerve endings might be, Pacinian corpuscles, Ruffini endings, and so forth. All right, question.
If you tear a ligament, are those like type 3 receptors permanently affected, or do they come back, or will you always have like decreased proprioception?
You will likely have some decreased proprioception in that specific ligamentous area, so you can try to tune up. You still have, those in other ligaments maybe nearby, or you can try to compensate with the other receptors either around that area or in joints above and below the injury. And if you really tear a ligament, you're always going to have some loss or compromise in your proprioception. That's why it's really important to do a lot of proprioceptive motor control stuff after injury, any kind of musculoskeletal injury. And now a lot of the conference stuff at CSM orthopedics was all about, what was the title of one? Your ACL has a brain problem, or your brain has an ACL problem, or something like that. It's like you cannot separate those out. So the motor control is just as important as the range of motion and the strength, of a joint. So that's because of some of these things that you lose. Okay, we get to go into the pathways now. So the first few are kind of a little more overwhelming because we got to lay the groundwork, get all the terminology, get all the different levels that we're going to be going up and talking about. But then it's all the same, kind of like once you know that. So the first one's going to take a little while, but then the other ones will go a lot faster. So just bear with me. We'll try to help you figure out ways to make this a little easier. It's not too bad if you just kind of take it one at a time and then look at the patterns. Okay. So now we're talking about that signal. So you've stimulated a peripheral receptor in the skin, in the muscle spindle, in the joint, wherever, a free nerve ending, a mechanoreceptor, a chemoreceptor. It's carried up on an axon in a peripheral nerve that could be a type 1, a type 1A, 1B, 2, A delta. It's coming up, then it eventually gets into a spinal nerve. So it goes from the peripheral nerve eventually all into the spinal nerve. Then it goes in the dorsal root, and then eventually into the spinal cord, and that's what we're going to do now. We're going to go up the spinal cord to wherever it's going to go from there to the destination it's going to go. So when you're trying to think about what's really important from a clinical perspective when you think about these pathways, is the main things you want to know is, first of all, what is the type of sensation it's carrying? What's the class of sensation it's carrying? And that's what these are going to tell you kind of right here at this level. I divided up by the type of sensation that these are carrying, okay? Does it cross the midline? When I say midline, the midline of your neuraxis, your brain, brainstem, and spinal cord there's midline right down the middle of that neuraxis. Does it cross the central nervous system at some point? If it does cross, where does it cross? Does it cross in the spinal cord or does it cross in the brainstem? Those are the two main areas it's going to cross, either in the spinal cord or brainstem. And then how will a patient present if they have damage to that pathway? If that pathway goes from the spinal cord through the brainstem all the way into the cerebrum, some pathways stop in a brainstem or they stop in the cerebellum. They don't go all the way up to the cerebrum. But if it goes to the cerebrum, you want to know if you have damage at the spinal cord level versus the brainstem level versus the cerebral level, how that might alter or change the symptoms that the person might have. Okay? So, what are the primary pathways for somatosensation? From a standpoint of type of sensation, we have what conscious proprioception. When I can conscious proprioception stuff, I'm consciously aware that my body's a certain position, my limb is moving in a certain way, and I can consciously tell you that, and I can think about it and feel that consciously. That's conscious proprioception. And discriminative touch. When somebody touches me in a very specific location, I can tell you exactly where that happened. Okay? That's one area. And that type of sensation is carried on two pathways. One for the body, which is called collectively the dorsal column medial lemniscal system, and then one for the head, called the trigeminal tract. this is called the trigeminal tract. It's coming through the trigeminal nerve. Okay, so conscious sensation of my face. You know, when somebody touches me or my jaw is in a certain position, knowing exactly where my jaw is and consciously being aware of that. Those are the two pathways. Central nervous system pathways. That's how that stuff gets to where it's going. Then we have this thing called the unconscious proprioception. It's our body awareness that we are not consciously aware of. This is all happening subconsciously in the cerebellum. When your muscle spindles and joint receptors are sending signals up, they're sending signals not only to your cerebrum that give you a conscious awareness of your body position, but they also send signals to your cerebellum. And your cerebellum can make adjustments to your muscles' function and so forth without you ever having to think about it. And that's why you can do a lot of things and move without thinking about it and make error corrections without thinking about it. That's all because of these unconscious proprioceptive pathways, collectively called the spinocerebellar tracts. We're going to talk about 4 of them, but collectively we're just going to call them the spinocerebellar tracts. Okay. It's nice that a lot of times the names will give you away some of the characteristics of it. Spinocerebellar. You know it's an afferent pathway because it's going from the spinal cord, spino, towards the cerebellum, spinocerebellar. Trigeminal tract, at least you know it's coming with the trigeminal nerve. Dorsal column, medial lemniscus system. Dorsal columns is the back of the spinal cord. It doesn't tell you exactly that it's going to go to the cerebrum, but that helps you identify where it's starts in the spinal cord, goes to the dorsal columns, and eventually in the brainstem it's called the medial lemniscus. It just changes names once it gets in the brainstem. It's called the medial lemniscus. Then we have pain, temperature, and coarse touch. Those modalities of sensation. Those are carried on for the body, the anterolateral system / spinothalamic tract. Okay. And then for the head, it's the trigeminothalamic tract. Spinothalamic starts in the spinal cord. It's going to eventually go to the thalamus, and if you go to the thalamus, you're going to go to the cortex because you got to talk to the thalamus to go to the cortex. Okay, so spinothalamic, trigeminothalamic. So that's going from the trigeminal nerve to the thalamus, and if you go to the thalamus, you're going to eventually go to the cortex. Okay. So that's the face. Pain, temperature, coarse touch from the face. Let's start real simple here. Sometimes I just like to have simple diagrams, can tell you more than looking at the fancy stuff and all the different levels first. So this is I think I included this in that little packet of information I handed out today. So let's go across. We're not looking at this one today. Here's pain and temperature from the body. This is anterolateral system / spinothalamic tract. Let's change the color a little bit. So let's say it's a free nerve ending out here somewhere in the periphery. It gets stimulated, sends a pain signal on a delta or C nerve fiber. Comes into the spinal cord on this side of the nerve axis. You've got the little dotted line that shows the center of our nerve axis or our midline. Comes in the spinal cord on one side. It crosses over that middle. And where does it cross? In the spinal cord, pretty close to where it comes in. Goes up the opposite side of the spinal cord. Goes into the brainstem. In the brainstem, this is the spinothalamic tract. However, in the brainstem, it's actually called the spinal lemniscus, but we'll get to that when we get to that. This goes to the thalamus, which eventually goes to the cortex. Okay, so thalamus and cerebral cortex. That's the pain and temperature pathway. Here is just pure proprioception. Okay, pure proprioception, fine touch. Discriminative touch and proprioception of joints. Okay, let's just call this one proprioception of joint movement and so forth. Next one's light touch. Information comes in to the spinal cord on one side of the body. It goes up the same side of the spinal cord. in the dorsal columns or posterior columns. Eventually it does cross over, but it waits to cross over until right at the bottom of the brainstem, right at that junction between the brainstem. It's right in the medulla that it crosses over, okay? And then it goes up to the thalamus and to the cerebral cortex. So this might be a Pacinian corpuscle or Ruffini ending or Merkel disc. This fiber right here might be a muscle spindle, and those fibers are A betas, 1Bs, 1As, or 2s. Okay, the type of axon goes in the dorsal column of the spinal cord, goes up on the same side of the central nervous system for a little while until it gets to the brainstem, crosses over, goes to the thalamus, which goes to the cortex. Light touch. Why light touch is not a great modality to test unless you do it is it's carried on both pathways. It's carried on the spinothalamic tract, which I'm pointing to here. This is just coarse light touch. And it's carried on the dorsal column medial lemniscal system. This is fine light touch. And unless you're very good about how you test that, if you just test light touch, you don't know what, if the person has, if you just do, like you just say, tell me if I'm touching your arm or your leg. They may have an intact anterolateral spinothalamic tract that gives them that coarse touch sensation, but their dorsal column may be messed up and you never tested that because you just tested this coarse light touch. You didn't test discriminative light touch, or you didn't test pain and temperature versus discriminative light touch. That's why it's important to do, if you're going to try to screen whether somebody has damage to the different pathways, it's best to do pain and temperature to test that spinothalamic tract, and discriminative touch, localized touch, or proprioception to test that dorsal columns. A lot of times it's just use vibration. Vibration is a very good way to test the dorsal columns as well. Coarse light touch doesn't tell you a lot because that's carried on both the dorsal columns and the, it just could give you confusing information. Last thing, unconscious proprioception. Those are the spinocerebellar tracts. We're going to talk about 4 of them. Information comes in from muscle spindles, joint receptors, so forth. It goes up the back part of the spinal cord in those dorsal columns, or not really dorsal columns, I shouldn't say. Some of it does. They do have some spinocerebellar tracts that go up in the spinal cord. We'll show you to them. They stay on the same side. Notice they never cross midline. And they go into the cerebellum. They never go up to the thalamus. They never go up to the cortex. Now, your cerebellum does actually send information that crosses midline up to that about what happened, what got there, but it doesn't directly get there. Okay, so it stays on the same side, and it stops the cerebellum. So most of your sensory pathways are contralateral. They cross midline, except for the cerebellar tracts. They stay on the same side. They're ipsilateral in nature. So we're going to go through these now in a little more detail. We're going to talk about conscious proprioceptive discriminative touch pathway first, which is referred to as the dorsal column medial lemniscal system. Because in the spinal cord, it goes up the dorsal columns, and once it gets to the brainstem, it changes name. Just like sometimes streets here in Dayton, you'll be going, once you cross one street, it's all of a sudden you're on the same street, but it changes names once you cross a certain street or something like that. This is what happens with some of our pathways. They change names a little bit once you cross into a new area. Like when you go from the spinal cord into the brainstem, this one happens to change names a little bit. We're going to start in the periphery and work our way up. So the types of receptors that contribute to this conscious proprioception, discriminative touch, pretty much everything contributes to that, even free nerve endings, okay? But it's more so those specialized, Meissner's, Merkel's, Pacinians, peritracheal, muscle spindles, GTOs, joint receptors, all that stuff contributes to conscious proprioception, conscious awareness of movement. The specific types of modalities, the sensory modality or experience that you get from all these receptors are these types of things. Pressure touch. How much pressure you're feeling at any given moment when you're interacting with an object, which that gives you an awareness of shape, size, and texture of an object. Kinesthesia is that awareness of body movement, okay, while you're actually moving, okay? It's this kinesthetic awareness of like, I can tell my elbow's extending or I can tell my elbow's flexing while it's happening. That's kinesthetic awareness, okay? Weight perception, how heavy something is sensed consciously. If you can feel consciously the weight of an object, this is by a combination of the modalities. It's the stretch that you feel in the muscle spindle, the ligament, the pressure in your hand if you're holding a bar. It's the combination of those modalities that tell you that's how much this object weighs, you know, in my hand or my arm. The positions, static position sense, okay? Spatial relationships with bodies and parts. Some people call that proprioception, but proprioception includes both static position sense and movement. Proprioception is more of a bigger umbrella term to include a lot of these things, okay? Proprioception involves kinesthesia, position sense, pressure sense. So really, proprioception would be the bigger catch-all for all these. And I'm getting more specific about the variations of types of awarenesses of sensory experiences. Then stereoagnosis is the appreciation of an object's three-dimensionality. So when I pick up my phone or I pick up a pen, I appreciate the three-dimensionality of it. Even if my eyes are closed, if you put a common object in my hand, I could probably tell you what it is just from my stereoagnosis. I could tell you if that's a cotton ball versus a penny versus a paper clip versus a pen if you put it in my hand. There's a very specialized region in the brain that takes some of that information and helps you develop that mental picture for it. And so that's what stereoagnosis is. And that's part of this conscious proprioception that we get. So here we go. We're going to go through the pathways here. And for most of these pathways, we're going to talk about the upper extremity versus the lower extremity, okay? And we're going to talk about orders of neurons. So there's more than one neuron involved in these pathways, okay? The pathways for sensation typically involve at least 2 orders of neurons. Many involve 3 orders of neurons. If they're going to go to your brain, your cerebrum, they're going to involve 3 neurons. If they're just going to your cerebellum or your brainstem, they may only require 2 neurons in the pathway. But we'll go through that. So this is a dorsal column medial lemniscal system. This is going to go to your cerebral cortex. This is going to go to conscious areas. This is going to take 3 neurons to get there, okay? So we're going to go through and talk about first order neurons, second order neurons, and third order neurons. These are the neurons in the pathway. So the first ones, we're going to always talk about a couple things about these. Where are their cell bodies? You know, and then where does it enter? Where does it ascend? Where is its synapse at? Okay? So let's start with the leg, and we'll just kind of follow one of these out. So first order neuron, let's start down here in the foot. So this is this person's left foot. This might be some Pacinian corpuscles, Merkel's discs, ligament receptors, whatever. They send a signal on this peripheral axon when stimulated. That peripheral axon could be a 1A, a 1B, a 2, an A beta. We don't know. It depends on the receptor that's stimulated in that foot. The cell body for that neuron is going to be in the dorsal root ganglion. So the cell body for the first order neuron is in the dorsal root ganglion, sitting right outside the spinal cord. It's going to go in the dorsal root, that back part of the spinal cord. And it's going to go through a very specific division of that dorsal root. The dorsal root can be divided into medial and lateral division. I'll show you a picture that shows you, but it comes in more medial, because it's going to go to that back part of the spinal cord. It's going to come a little more medial to the middle part of the spinal cord. It's going to go up those dorsal columns, okay? It enters, so that's where it enters the spinal cord. Then it ascends up the back part of the spinal cord as in one of the dorsal columns. I said, you hear dorsal columns? There's 2 columns there. It sends in the posterior funiculus. The funiculus is the big back part of the whole back part of the spinal cord as either the fasciculus gracilis or the fasciculus cuneatus. If it comes in below T6, it's coming from the lower body. It's going in and it goes up the dorsal column called the fasciculus gracilis. Remember we said gracilis muscles in the lower body, so that's how I remember that. If it comes in above T6, it joins in the dorsal columns a little more laterally because it layers onto it as part of the funiculus cuneatus. It goes up to the lower part of the medulla. So you're going to have to start getting yourself familiarized. So this right here, this cross section right here, this is a horizontal section through the lower part of the medulla. Don't have to worry about all the detail in there yet, but you have to start worrying about a few things. So let me just erase that. You have to worry about these two little guys back here. You have to worry about a nucleus gracilis and a nucleus cuneatus. Okay, the more medial one is nucleus gracilis. The more lateral one is nucleus cuneatus. Okay, so we're going to worry about right now. This is lower body, right? So lower body is going to come up. It's going to enter in this whole region back here is the posterior funiculus of the spinal cord, but it's going to enter in and it's going to go up what's called this little region right here. It's more medial. It's called the fasciculus gracilis. So this little area here is the fasciculus gracilis. It's going to go up the spinal cord all the way up through the thoracic and cervical spine as the fasciculus gracilis. That axon's going up, and then it's going to eventually go to the nucleus gracilis, where it's going to synapse with another neuron, the second order neuron. So the cell body for the second order neuron, the next neuron on the pathway, the cell body, is going to be in that nucleus gracilis. Immediately after synapsing with that cell body in the nucleus gracilis, the cell body projects an axon that crosses over midline at this point. It projects that axon medial and it crosses over right at that same area in the lower medulla, and then starts to ascend again another little region in the medulla right here, this little flattened area that now is called the medial lemniscus. Because remember, lemniscus is a ribbon-like structure. It's a little more flattened. The dorsal columns are a little more columnar in nature and a little more round in nature. Now it's flattened into this meniscus, or lemniscus, I'm sorry. And then now the lower body conscious proprioceptive information is going to be carried through the brainstem in the medial lemniscus. And it goes up this way, and then it actually turns more, here it's more arranged anterior to posterior, here it's more medial lateral, then here it gets a little more anterior to posterior again. Because what happens is you've got this, so here's my medial lemniscus. My hands are the medial lemniscus on each side. They actually do this. That's, as it goes up the brainstem, those paths twist like this. They kind of twist as they go up. And it's like a ribbon twisting. If you take a ribbon and kind of twist it, that's what those axons are doing. They're just kind of twisting as they go up through the brainstem. Eventually, that axon is going to project to the thalamus, and it's going to make a synapse with the next neuron, the third order neuron in this pathway. And that cell body for that third order neuron is located in the thalamus, and it's located in a very specific region of the thalamus called the ventroposterolateral part of the thalamus, the VPL, the ventroposterolateral thalamus, the VPL. Then from the VPL, that neuron cell body in the VPL is going to project an axon to the appropriate region within the brain. Now this picture actually is wrong. It's showing that red, which is coming from, oh no, that is not wrong. I'm sorry about that. What's color is this down here? Is that, that's a green, right? Yeah, so that's right. So that's going right here. The lower body leg is coming over here to the homunculus, or to the post-central gyrus that's on that medial surface. Remember we said that the leg kind of goes over the wraps and is over on the medial surface of your cerebrum. That post-central gyrus that extends all the way over to the medial surface of your cerebrum. Most of the post-central gyrus we see on the lateral surface. That's mainly face, hand, upper body, trunk. If your leg goes over the medial surface, and we'll show you the homunculus here so you see that later. Um, but that's projecting to that specific region. Got it, sort of? Now let's just follow upper body real quick. Talk it through again. So here's a hand. Whatever. Pacinian corpuscle, Merkel's disc, Golgi tendon organ, muscle spindle sends information via the appropriate axon. Cell body, dorsal root ganglion. It's entering in the cervical spine, right? Because this is a hand. It might be at the level of C6, C7, or C8 right there, okay? It's coming in later, and so it adds on. So it comes in here into the dorsal columns. It comes into the dorsal root, goes in the dorsal column here, but it's added on laterally to the dorsal column as the fasciculus cuneatus. The fasciculus gracilis was already there, right? Because from the lower body, then the cervical, the upper body stuff gets added on and layered on. So down in the lower body, you only have a fasciculus gracilis. It's not until you get into the cervical spine above T6 where you start to have a fasciculus cuneatus. Because that's now your upper body and upper trunk stuff getting added on. Okay. So that's that. Goes up to the nucleus cuneatus. Synapses with the cell body in the nucleus cuneatus. That axon projects and crosses over right at that level of the lower medulla. That's called the decussation there. They call it the lemniscal decussation. It's a decussation of the medial lemniscus because it's crossing. Because you have information coming from this side, it's crossing over this way. Okay, from the other side of the body. And then it ascends in the medial lemniscus with the rest of your body. Medial lemniscus all the way up to the thalamus. Same area in the thalamus, the VPL, ventroposterolateral nucleus. Synapses with the third order neuron. And that third order neuron projects to the appropriate region. The hand would be projecting to this region up here in the homunculus on the post-central gyrus, sorry. Primary sensory strip, post-central gyrus. I've already mentioned this. So that third order neuron coming from the thalamus up here, the place it ascends to this area right here, this region right here, that's the internal capsule. That's where you have a lot of those white matter fibers. You have both motor and sensory fibers running through that there's a little condensed region that's right between your thalamus and your basal ganglia structures where things get narrowed down. That's the internal capsule. That's where those axons from the third order neurons are running through that internal capsule region before they splay out to go to their final destination on the cerebral cortex. And they terminate in that post-central gyrus. Feeling okay about that? All I want you to appreciate from this picture, this is a similar picture showing, here's a lower body's in blue in this case, upper body is in red. This is spinal cord, these first two sections, then we have lower medulla and upper medulla, pons, and brains and midbrain. Just notice how when those come in, the red and blue, so it starts out you only have blue, right, down lower because that's lower body. Then you add the red on to the outside. That's when you start adding on that fasciculus cuneatus. Then here's where you get up into the lower medulla. Those both cross over to the other side into that lemniscus, that medial lemniscus. But you'll notice how it stays somewhat somatotopically arranged. So you see that the red here is more posterior and the blue is more anterior when you're in the lower medulla. That as it's going up, it starts to twist. You'll notice here in the pons that the red now is more medial and the blue is more lateral. So the fibers carrying your proprioceptive information from your upper body, in this case, are more medial, and in this case, the lower body is more lateral. And then it keeps twisting a little bit, and up here now you get more of an anterior-posterior arrangement again, where the lower body is more posterior in the midbrain, and the upper body is a little more anterior in the midbrain. Okay, and then when you get into this whole region right here, this is the thalamus. This whole region of the VPL it has a region that's more associated with the lower body, I mean upper body in red and the lower body in blue. I say that because you can have a very specific stroke possibly that affects the VPL and it hits just the part of the VPL that's associated more with upper versus lower body. It maintains some somatotopic arrangement throughout. They don't totally mix. Do you see what I'm saying? Those fibers stay a little bit separated, and it's sometimes it's important to know that because of lesions affecting certain regions could lead to more upper versus lower body involvement. Most of the time, it knocks out the whole thing because these are tiny little structures that are really close to each other. But sometimes it's just, I just want you to aware that there is some we call somatotopic arrangement of these fibers all the way up to the back of the spinal cord, all the way through the brainstem, that they stay sort of separated, near each other, but separated and stay. And then they eventually, where they really start to separate is in the cerebral cortex. That's when they get far apart from each other. That's why your leg is way over on this side of the middle part of my brain. My hand is way over on that side. But down lower, they're closer to each other. So all I'm saying is, like, if you've got a stroke and their whole side of their body is numb, their arm, their leg, their face, you're probably in a region where they're all close to each other. And the place where they're all really close to each other, the most common place for stroke, is the internal capsule. You know that internal capsule? That's where everything is near each other. That's where your motor, your sensory systems for your whole body are really close to each other. So internal capsule strokes are really bad. If you have a stroke more on the outside or the surface of your brain, it might affect your arm and your face, but not your leg so much because your leg's way over here. You see what I'm saying? But if you have a stroke deep in that white matter in the internal capsule where a lot of little pathways are really close to each other, you can have a pretty small stroke that causes devastating consequences. This is showing, so this whole thing that I'm circling there, let's assume this whole thing is your dorsal root. We're looking at like a blown up dorsal root, okay, where all these axons of sensory axons are coming in the back part of your spinal cord. We can divide that dorsal root into a medial group and a lateral group. The medial group is going to go right here into this dorsal column region, okay? That's where your 1As, A betas, your 2s, your fast proprioceptive fibers go in that medial division, more medial, because they're going to go dump into that dorsal column and go right up. Where your lateral group, right here, more lateral, it's more lateral in nature, it's going to go in that dorsal root more laterally, but it's because it's going to actually go right into the dorsal horn here because it's going to make a connection with cell bodies right here. That's the C's and the A deltas. Those are the pain and temperature fibers, okay? They're coming in just a little more laterally in that dorsal root. Does that make sense? This is just a cross-section through a spinal cord. Here's your dorsal. That's your dorsal horn on each side. That's a gray matter. It looks lighter here because it's the way they stain the spinal cord. It's opposite. This is your this whole thing right here is called your posterior funiculus of your spinal cord, which we actually, if you look closely, we can divide it into these 4 areas. There's a little bit. This is your fasciculus gracilis. This is your fasciculus cuneatus, and then same on the opposite side. Fasciculus cuneatus, fasciculus gracilis. That's the back part of the spinal cord. So this would have to be above T6. If you have both the fasciculus gracilis and cuneatus, you have to be above T6. Oh, I'm sorry. Yep, I always do that. Yes, sorry about that, guys. Yep. This is fasciculus gracilis, fasciculus cuneatus. Notice how fasciculus gracilis is smaller than fasciculus cuneatus. Why do you think that is? It's physically smaller. The real estate it takes up is smaller. You actually have a lot more sensation and stuff coming from your upper body than your lower body, so it actually takes up more volume because you have all those little receptors and neurons coming from your fingertips and everything. You have these big receptor fields and less, discriminative information. Your lower body isn't for manipulating objects. It's for gross movement and just some sensation of general pressure from the bottom of your feet, where your upper body has much more fine, it's much more richly innervated sensory-wise, so it's going to take up more space. There's more axons coming from your upper body. That's why the fasciculus cuneatus is bigger than the fasciculus gracilis. This is that one on your lower left, that's a cross-section through the low medulla. Actually, it's a cross-section through the medulla at the level of the nucleus gracilis and nucleus cuneatus. So this little area here, this is the nucleus gracilis. This is the nucleus cuneatus. So again, these are reverse images here. So whatever's dark is actually white matter. Whatever's light is actually the gray matter. And so these are the cell bodies. These are where the cell bodies are going to be for your second order neurons right here. Their axons are going to project and they're going to cross over. So we're going to cross over midline here. And then right here and here, this is where your medial lemniscus starts. So this is going to cross over here, and then once they cross over, they're going to go up from here, okay? You have, what's happening here is here's your dorsal columns are coming at the back of your spinal cord. We're going to kind of make hashtag. They're going to come out right here. This area back here, this is your fasciculus cuneatus. This little region back here is your fasciculus gracilis. So the axons are going to project up from the spinal cord, and they're going to come up and they're going to communicate with the cell bodies here that are located in the nucleus cuneatus and gracilis. And that signal is going to, those fibers are going to come over here and they're going to cross over, and then they're just going to be going straight up the brain stem all the way up. Yep. Yeah. They cross in the lower medulla. The actual crossing point or location specifically is the lemniscal decussation, which is in the lower medulla. And so the lemniscal decussation is this region, right? So you got, these are crossing over. So you got these crossing over from this side, and then these guys over here are crossing over from this side. So this region right here, that's the lemniscal decussation. That's where you're getting the crossing fibers from each side of your body that are crossing over to go to the opposite side from your dorsal columns that are now turning into your medial lemniscus, but they're all crossing over before they get a little more flattened in arrangement and become that ribbon that goes up through your brainstem to go to the thalamus. This cross section looks kind of like a upside down butterfly or whatever. That's a midbrain. You'll see that. You'll automatically know that's the midbrain. This is actually one of your colliculi back here. Remember the colliculus, those little bumps on the back of your brainstem? This is the front part. This is the cerebral peduncle area, front part of the brainstem or midbrain. But anyway, this right here is your medial lemniscus. Is that region right there. Again, it's the opposite. The dark stained stuff is white matter. Those are axons. That's the medial lemniscus. Okay. Back here, looking at the external features, there's a bump here and a bump here. Medial, those are your gracile tubercles. And right, if you poke a pin in there or cut right into there, right underneath that, it's going to be your nucleus gracilis. The nucleus gracilis is sitting right underneath that gracile tubercle. That's what's making it bump out there a little bit, because there's some cell bodies that are grouped there that are making a little bump right there at the back. So the external feature, it's called the gracile tubercle. The internal feature is the nucleus gracilis. If we cross-section that, we did a cross-section right through there and looked right down on top of these, that's where your nucleus gracilis would be. And then there's a bump right here and a bump right there. Those are your cuneate tubercles, because right beneath those is your nucleus cuneatus. So we have a bump here and a bump there. Those are your gracile tubercles, and then your cuneate tubercles. And then we have these are called the same thing, whether you're looking at it from the back or in cross section. This is the fasciculus cuneatus, more lateral, than the one here, more medial. That's the fasciculus gracilis. So it's called the same thing from the external feature side of it, or so if you cross-section horizontally and looked right down, it would still be called the fasciculus gracilis and fasciculus cuneatus. Got it? This is just a thing that I made up for you can draw on yourself to do some of these. Let me just draw this one real quick to show you an example. Let's do the 3 orders of neurons of different colors. Let's start with red. Dorsal column, medial lemniscal system. This is let's do a leg. So we've got our nervous system here. We've got our neuraxis is this dotted line in the middle. That's our center of our nervous system. Then you see I have it divided up into a lumbar spine, cervical spine for the spinal cord. Then we get into the medulla, pons, midbrain. We have a cerebellar hemispheres on each side, right there, that junction between the medulla and the pons. And then we have a thalamus divided up into a medial and a lateral part, okay? And then our cerebrum. So if we do our lower body, dorsal column, medial lemniscal system. So here's our little specialized receptor out here. We stimulate it. A lot of times in those, let's say this is a Pacinian corpuscle, this is going to happen to be an A beta fiber that's going to send that on. It's a pretty decently fast, not super fast, but decently fast myelinated fiber, okay? This is, before it goes into the spinal cord, this little guy right here, that's cell body, that's our DRG, dorsal root ganglion. I don't know, this may be L5, whatever. That comes in the spinal nerve root of L5, goes into the dorsal root, medial division of the dorsal root, enters into the dorsal column in the lumbar spine, and it goes up the same side. And in that dorsal column here, it's going to be called the fasciculus gracilis, right? So fasciculus gracilis. It goes up, stays the fasciculus gracilis in the spinal cord. Now we get to the medulla right here, low medulla. Now we're going to change colors. Let's go to blue. So this is the first order neuron. Now we're going to synapse. Where are we synapsing? What's it called? What's that area called? The location called? The nucleus gracilis. So this synapse is the nucleus gracilis. That second order neuron is going to cross over immediately to the other side of the low. This is kind of low medulla. We're in here. And then it's going to go up, and it's going to go up as the medial lemniscus, right? So now it's called the medial lemniscus here. It's going to stay the medial lemniscus all the way up. It's going to go to the lateral part of the thalamus, the VPL, the ventral posterolateral part of it. So it's the kind of underneath surface, the back part of the thalamus, and the lateral part of the thalamus is that back lateral part, underneath surface of the thalamus, ventral posterolateral part of the thalamus. Let's just show it going to this lateral part, and then let's change it to a green. It's going to synapse with the next cell body, the third order neuron in the VPL. So this right here, this location is the VPL. And then this is coming from the lower body, so it's going to project to the homunculus over here, to the medial surface of the post-central gyrus, okay, because that's where the leg and foot are more over that medial surface. Then if we did the upper body, it would just be coming in the cervical spine. It would be doing the fasciculus cuneatus, nucleus cuneatus, and it would go project over to more the lateral hemisphere. Got it? So the first one is the hardest one, because then all the others, we talk about similar locations and regions, so we can just kind of move through other ones. Let's just do trigeminal pathway, and we'll take a break. So trigeminal pathway is the equivalent, but it's from the head. Okay, pain, temperature. This is conscious proprioception. Trigeminal pathway, conscious proprioception from the face and jaw. Okay, mandible, jaw region. First order neurons. Cell bodies are in the trigeminal ganglion. So here's that trigeminal ganglion. All right, let me change this back over to red. So here's the cell body. Again, this is coming from the face, and this could be A betas, 1As, whatever, if it's a muscle spindle versus a joint receptor. Okay, temporal mandibular joint, coming from that. Facial, skin, muscles. So this is the equivalent. We have a cranial nerve ganglion here called the trigeminal ganglion. That's the equivalent of a dorsal root ganglion down in your spinal cord. It comes in at the pons. That's the pontine levels where that exit in that basilar pons, that front part of our pons, that's where the trigeminal nerve comes in. It pretty immediately synapses with a nucleus called the trigeminal main sensory nucleus. Cranial nerves are a little bit unique. Especially the trigeminal nerve, it actually has 3 separate nucleuses that subserve the different sensory functions and motor functions of it. So this actually has 4, but this is called the trigeminal main sensory nucleus. That's going to be the first. This is the first order neuron. It's going to synapse with the second order neuron in the trigeminal main sensory nucleus. That axon from that second order neuron is going to project across midline into what's called the trigeminal lemniscus. Hey, that's kind of nice and convenient that it's similar. The medial lemniscus is from the body. The trigeminal lemniscus is for the face, and it also gets a little more flattened in nature there. And the trigeminal lemniscus actually ends up joining up right at the tip of the medial lemniscus. It actually joins up with your medial lemniscus, which is nice. And then it's kind of, once you get up at this, so this is the midbrain region. Once you get into the midbrain, it's right here at your medial lemniscus is here, and it's right at the tip of your medial lemniscus. It joins up with the medial lemniscus. So the sensory fibers for your face join up with it. It goes to the thalamus, but it goes to a slightly different part of the thalamus than the body. It goes to the ventroposteromedial part of the thalamus as opposed to the lateral part. When I think of all the information, sensory information, pain and temperature, proprioception coming from your face, if it's going to go to your cortex for conscious awareness, it's going to go to the thalamus. It's going to all go to the VPM, the ventroposteromedial part of the thalamus, where your body all goes to the VPL. Okay, I think of the VPL, leg is an L, and that makes me think body. VPM, I think mandible or mastication. That's why I think VPM is associated with face. That's what helps me remember it. So that just goes to the thalamus, just right next to the VPL is the VPM. Okay, and then it goes from there. The third order neuron then goes up through that same internal capsule region, but in this case, it goes farther down on the homunculus, down more laterally and farther down in that postcentral gyrus to the region that's where your face is. That's where the facial area of your brain recognizes that. Okay? So that's just called the trigeminal pathway for conscious proprioception from your head and face. A little simpler. This is just showing you I'm not going to worry about the detail there on that. When we get to cranial nerves, we're going to go into more detail on each of these cranial nerves and all those. So let's take a break, come back, we'll get through unconscious proprioception. All right, so we just talked about conscious proprioception from the body and the face, right? So this is like I know what my body's doing unconsciously. Now this is unconscious. You have no awareness of this. This is all happening in the cerebellum, okay, which you don't have conscious awareness of. And we're going to do a whole lecture on the cerebellum. This is not about the cerebellum. This is just how information gets to the cerebellum. That's all we're worried about right now. We'll leave that for another day of what happens to the information once it gets to the cerebellum and how it's processed. But we're just talking about how does it get there in the first place. So collectively, these are spinocerebellar tracts. Starts in the spinal cord, goes to the cerebellum. So, unconscious proprioception. We have spinocerebellar pathways. There are two, we have four of them. Two of them are what's called high fidelity pathways that actually bring in the proprioceptive information from the actual extremities, limbs, about what's happening in those muscles and tendons and joints. That's bringing in lots of high fidelity information, okay? And they start out in the periphery, in your joints. We have two pathways that are called internal feedback tracks. So they're kind of unique. They actually start within the actual spinal cord itself. They're monitoring information that's coming in from those peripheral areas right at the spinal cord level, what's happening at the spinal cord level in each level. So they don't receive information directly from the periphery. They're monitoring neuronal interneuron activity when the information first comes into the spinal cord. So they're internal tracks. They're not starting out in the periphery. So the high fidelity pathways, the posterior spinocerebellar and the cuneo, are for bringing in the information from the discrete muscle spindles, ligament receptors, joint receptors from the periphery. The internal feedback pathways are just within the spinal cord up to the cerebellum, okay, that are monitoring what's happening, some activity in the spinal cord region. So receptors, muscle spindles, GTOs, joint receptors. Those are the ones that provide information to these spinocerebellar pathways. Probably there are some information that come from some of the other, like Pacinian and Merkel's and Ruffini's, but most of it is coming from these more deeper sensory organs, you know, spindles, Golgi tendon organs, and joint receptors. The modalities, they understand your posture, posturing, movement of lower, this is spinocerebellar pathway we're talking about first, okay? So what it's telling you is about the posture of your lower body and position of your lower body unconsciously, and how your lower body is moving at any given moment, okay, unconsciously. So let's talk about the posterior spinocerebellar pathway. Since it's not going to the cortex, it only has two orders of neurons. It doesn't have to go stop by the thalamus, so it's just going to the cerebellum. It stops off early, so it's just got two orders of neurons. So here is a little squiggly thing representing a muscle spindle in a quad, okay? So this comes in, it's got a peripheral axon here. If that's a muscle spindle, that axon is either a 1A, a 2, or a 1B. I don't know for sure, or all the above, okay? So that axon, well, it's got to be on one or the other, but you're going to have 1As, 2s, and 1Bs coming from those intrafusal fibers in your muscle spindle, okay? They're going to come in the back part of the spinal cord to the dorsal roots, and the cell body for that axon is going to be located in the dorsal root ganglion at the appropriate level. If this is a quad, it might be L2, 3, or 4, or something like that this is coming in. It comes in through the medial division of the dorsal root, just kind of like your conscious proprioception comes into the medial division of that dorsal root. It makes its first synapse in a region of the gray matter. This is that posterior horn. There's a little region that's more medial over here. It's a little differentiated in its cell makeup, and it's called the nucleus dorsalis, or Clark's nucleus, okay? And that Clark's nucleus is only from T1 to L3. If you have unconscious proprioception coming in from down in the foot, down here lower than L3, the axon will go up in the dorsal column in the fasciculus gracilis up to that L3 region to synapse with Clark's nucleus, okay? If it comes in at L3 up to T1, it'll go right in and synapse with that Clark's nucleus, which is a column of cells throughout that gray matter in the spinal cord that's differentiated and called Clark's nucleus. That's just a specialized region of that dorsal horn. That's where that synapse is, that first synapse for that. So erase some of this stuff. So that's this region right here. And that Clark's nucleus extends up and down from T1 to L3, okay? So if you come in though below L3, if you're an axon that comes in below L3, down in L5, S1, it's going to go up in the dorsal, in the fasciculus gracilis, up to L3 or L2, and then it's going to synapse with Clark's nucleus for its second order neuron. Does that make sense? Once it makes its synapse, that axon is going to swing laterally, and it's going to go into, this is a region, this whole area here is called the lateral funiculus, but it's going to go into a specific region right here that's called the posterior spinocerebellar tract. Because right in front of it is the anterior spinocerebellar tract, but there's a little region here called the posterior spinocerebellar tract. And that posterior spinocerebellar tract is going to stay posterior and lateral. It's going to stay in the same place in the spinal cord here. And it's going to actually stay in the same region, this posterior and lateral region, up in the lower medulla. And then it's going to go up and enter in to the cerebellum through the inferior cerebellar peduncle. Remember the little connecting stalks or parts to the cerebellum? The inferior cerebellar peduncle is how things from below the cerebellum tend to get into the cerebellum. So this is these fibers, these axons coming up from below the cerebellum are going to go into the inferior cerebellar peduncle and then go into the cerebellum. And then communicate with the appropriate region in the cortex of the cerebellum associated with the lower body. Okay. So that's the posterior spinocerebellar tract. High fidelity pathway bringing in lots of information from muscle spindles, joint receptors from the limb. And this is from the lower body, posterior spinocerebellar tract. Got it? Just two orders of neurons. All I wanted to show you here, this is a cross-section through a spinal cord. This is a cervical spinal cord because you can see we have this is a exaggerated fasciculus, cuneatus, and gracilis back here. But they're showing this region here, this medial kind of region right here. We have lamina that are kind of differentiated functions on one side, but then you have nuclei that are specialized groups of cells that are not arranged in a lamina. Those nuclei are over here too. They're just showing you that you have both a laminar kind of differentiation in your gray matter, but you have a nuclear differentiation too. So don't worry about the laminar part right now. But there is a specialized group of cells right here called the nucleus dorsalis, or the dorsal nucleus, or Clark's nucleus. That's associated with that's where the second order, the cell bodies for the second order neurons lie from L3 to T1 for posterior spinocerebellar pathway. Okay. So if you're coming in right at this level and you've got the nucleus dorsalis, you can go and you can synapse with it. This will swing lateral, and then right here is the posterior spinocerebellar tract, and we'll go up this posterior spinocerebellar tract. But if you're coming in below, and actually this would not be a cervical. This would have to be like a T5, 6. This couldn't be cervical because your nucleus dorsalis is only from L3 to T1. So this has to be like T1, T2, T3. Because it has to be T6 or above, but it has to be between T1 and T6 because we have fasciculus gracilis and cuneatus that are both showing there. So, but if you come in from below, you're coming in like down at S1, L5, you actually have to go up the dorsal column in the fasciculus gracilis before you then synapse with nucleus dorsalis at L3 or above. Okay? And then swing your axons laterally to this posterior spinocerebellar pathway. Then that just stays in this posterior spinocerebellar tract all the way up through the brainstem into inferior cerebellar peduncle into the cerebellum. Notice though that it all stays on the same side. It's ipsilateral, never crosses. So the left side of the cerebellum sensing is bringing in information from the left side of the body. Let's talk about the upper extremity equivalent of that, the cuneocerebellar. And if you remember that the cuneate nucleus, that's also upper body, but conscious proprioception. So that's kind of how I remember the cuneocerebellar tract is the upper body equivalent for the spinocerebellar unconscious proprioception. Cuneocerebellar is how I kind of remember that. Same types of receptors, muscle spindles, Golgi tendon organs, joint receptors from the upper body, same types of modalities of sensing your position, movement of the body, but unconsciously. So again, since this does not go to the cortex or the thalamus, it only has two orders of neurons, first order and second order. Cell bodies are in the same spot as everybody else, dorsal root ganglion. It also enters the medial division of the dorsal root. It's going to actually ascend in the fasciculus cuneatus. So it's actually going to run up initially with your conscious proprioceptive fibers in the fasciculus gracilis. Okay, so here's your upper body, a little muscle spindle in the bicep, comes in, dorsal root ganglion, comes in the medial division of the dorsal root, and it's going to go and join the fasciculus gracilis. It's going to go up the fasciculus gracilis, and it is going to make its first synapse in what's called the lateral cuneate nucleus. There's a nucleus that's even more lateral than the cuneate nucleus. So here's the cuneate nucleus right here. Cuneate nucleus. This is the lateral cuneate nucleus. That's the gracil nucleus, or nucleus gracilis right there. So there's actually another nucleus a little more lateral called the lateral cuneate nucleus. This is for the unconscious proprioception of the upper body, where the cuneate nucleus is for the conscious proprioception, right? So it's going to go up it's going to make its synapse with the lateral cuneate nucleus, and then it's going to send the next axon projection from the second order neuron, ascends through the posterior medulla as the cuneocerebellar tract. Once it makes that connections, it's the cuneocerebellar tract, okay, through the rest of the brainstem before it gets to the rest of the medulla, before it goes in through the inferior cerebellar peduncle on the same side. So ipsilateral pathway, inferior cerebellar peduncle. That's the same as we saw with the posterior spinocerebellar pathway. Does that make sense? Cuneocerebellar, unconscious. Now, we're going to talk about the internal feedback pathways in the cerebellum. These are the ones that don't have direct information coming from the outside. They're actually monitoring activity at the interneuronal level at individual spinal cord levels, because you have converging information at the spinal cord level. What you have is you have descending motor signals coming down. You have incoming sensory signals coming in. They merge and meet right there at that spinal cord level, and you have a spinocerebellar tract monitoring the match between the motor signal and the sensory signal to see if they match up appropriately for the activity you're doing. If they don't match up, they send a feedback back to the cerebellum and say, you better make an adjustment, because there's not a match between the motor signals coming down and the sensory signals coming up. They can actually assess and monitor the activity at the interneuronal level at each spinal cord segment. Okay, so they indirectly get information from those last pathways we just talked about, because they get to kind of like sense the information as it's before it even gets up to the cerebellum. Okay, so it's kind of like they're in a very ideal position to monitor both motor signals you're sending down to create movement, but also monitoring signals coming in about what actually occurred out in the actual movement that you created by the sensory feedback. Okay, so that's what they're doing. We're not going to draw it out there, but let's show anterior spinocerebellar tract. So the anterior spinocerebellar tract is in the cell bodies are in the gray matter of the thoracolumbar spinal cord. So this is the anterior spinocerebellar tract is the internal feedback loop for the lower body. Okay, so it's in that white matter in lamina 7. I don't know if I have a picture of going back here. Oh, right there. This is lamina 7. This whole region right here is lamina 7. This area right here, lamina 8 and lamina 9, this is where a lot of your motor neurons are. And you can see how it's perfectly located here near, like Clark's nucleus, near your motor neurons, that it can sense information both coming down from motor neurons and information that is coming in near that same region. So it's in that gray matter in the spinal cord, kind of in this ideal central location to monitor both sensory information coming in from this dorsal horn region and motor information that comes down to these anterior horn cells and motor that's more in that anterior horn. It's kind of located in that gap between the dorsal horn and the anterior horn to monitor information coming from to both of those regions. Okay, so what it does, it sits right here. So here's that lamina 7. It's sensing information coming in from here. It's sensing information coming down from motor neuron. I want that right. Stop writing. Did it lock up on me? It did. My pen locked up on me. That's not good. Well, I need to be able to point stuff out. Follow my cursor here as best you can. So there's the cell body for it. It crosses immediately. Crosses midline in this thing called the anterior white commissure. There's a little bit of white matter right there, right in front of that gray matter. There's a little bit of a white matter, a little tiny thing called the anterior white commissure. Crosses over the axons and then it goes into the anterior spinocerebellar tract, which is right in front of the posterior spinocerebellar tract. Just a little anterior to that. And it stays as the anterior spinocerebellar tract all the way up through the brainstem. But what's interesting is it goes in through it skips going through the inferior cerebellar peduncle like most of the information, and it goes through the superior cerebellar peduncle. But what you notice is it crosses, most of it crosses back over. There's a few bits of information that stay on the opposite side, but it crosses back over and ends on the same side it started. So it crosses over twice. But since it starts on one side, it ends on the same side, it's still considered a ipsilateral tract because it crossed twice. However, I will say there's a few fibers that do cross over, but we're going to call all the spinocerebellar pathways as ipsilateral. Even though this one crosses over once in the anterior white commissure, ascends, and then crosses back over to the other side of the same side of the cerebellum that pathway started on. That's why that says recrosses mostly. You know, there's a few fibers that stay contralateral. So that's the anterior spinocerebellar tract. Then we have a rostrospinocerebellar tract, which is now the upper extremity, internal feedback. That's kind of a equivalent or helps with the cuneo-spinocerebellar system or tract. So same kind of things, no receptors. It monitors the activity of spinal interneurons. Pen's still frozen, so we're going to follow my cursor here. So muscle spindle in the bicep. Actually, no, we're not following that. Sorry. We're starting right down here. Cervical cord. We're right here in that lamina 7. Monitoring information between sensor information coming in and motor information coming down, saying if there's a match between those signals and if we need to make any adjustments. It goes up in the spinal cord in the posterior medulla. That's all you got to know. Just goes up somewhere in the back of the medulla there. And you can see it right here. It's just there's some white matter here that are back in this area, in the back part, just posterior medulla. It enters the ipsilateral cerebellum. This one actually just goes through that same inferior cerebellar peduncle and into the cerebellum. terminates in the cortex of the cerebellum. Do you think I really care whether it enters the inferior versus superior cerebral or peduncle from a clinical perspective? I think it's good to just be aware of it, but I'm not going to try to trip you up on stuff that's not really clinically relevant. The clinical relevant stuff about this is that, yes, there's some information that's coming from external receptors that are unconscious in nature, via the posterior spinocerebellar and cuneo-spinocerebellar tracts. There's some internal feedback pathways that are also monitoring spinal interneuron activity, via the rostrospinocerebellar tract and the anterior spinocerebellar tract. But all of them start on the same side, and all of them ultimately end on the same side, and they all just tell us about unconscious proprioception. So what that means is if I have damage to my cerebellum on my right side of my cerebellum, my right side of my body might have poor coordination because it's the same side. Versus like conscious proprioception, if I have damage to the left side of my brain, I'm going to have loss of conscious proprioception on the right side of my body because they cross over. Those are those are some of the key clinical things to proceed with this without getting lost in some of the detail. It's just good to hear it once and know the terminology so you can be a more informed reader when you're reading research and you're reading textbooks and things like that. You just don't feel overwhelmed. You can interact with that material, but those are the clinical things to get out of it. All right, we're going to get through pain, temperature, and coarse touch. This one will be pretty easy compared to all the other stuff. So this is the anterolateral system or the spinothalamic tract. So some people use those terms interchangeably, but I don't think it's great to use those terms interchangeably. Anterolateral system encompasses, I consider, kind of all the pathways that carry pain and temperature. I think of the spinothalamic tract as the one that's carrying specifically fast pain from the body. But just know that some textbooks and some people will talk about these interchangeably. To me, the anterolateral system contains the spinothalamic tract, but also contains all the other pathways initially in the spinal cord. Okay. Nice part about these pathways we're going to talk about all start in the same locations, all start out going up the same area of the spinal cord, just that they all end up terminating a little different spot. Okay. So this is not how you want to do your sensory exam in the clinic. Although some of your patients have such poor sensation that you literally probably could do that to them and they wouldn't be too bothered. Real bad peripheral neuropathy. So the anterolateral system, as a system, contains all these. The spinothalamic, spinolimbic. I noticed that they tend to change some of the names on these editions. So now the 5th edition, I don't know if the 4th edition calls the spinolimbic, they call it the spino-emotional pathway versus the spinolimbic. So they stopped calling it spinolimbic. But the limbic system is associated with emotion. So spinal-emotional or spinolimbic. I will probably stick with spinolimbic, but I've changed even on this little chart. I think I added emotional, yeah, and on this one too, the table I added spinal emotional versus spinal limbic. Okay. So spinothalamic is fast pain and temperature from the body. Spinal limbic is slow aching pain. Spinal mesencephalic is also slow aching pain, and spinal reticular is also slow aching pain. They just all end up at different places. This is actually really pretty easy. Follow my cursor. Spinothalamic. You know that one starting in the spinal cord, it's going to go to the thalamus, and if it goes to the thalamus, it's going to the cerebrum. So that's that fast pain going up to my cerebral cortex. It tells me, hey, it hurts right here, okay, because I can localize that if it goes to my cortex. Spinal limbic, it's going somewhere to my limbic system. It's not going to even not going to go to the thalamus. It's going to go to the well, this one does actually go to the thalamus and then to the limbic system, but it's going to go to the limbic system, which is associated with your emotional responses to pain, okay? Spinomesencephalic. Remember, we know what the mesencephalon is, right? That is the midbrain. Remember, the midbrain stays in the mesencephalon. We have mesencephalon, myelencephalon, mesencephalon, metencephalon, and myelencephalon. The mesencephalon is just the midbrain. So that's going to the midbrain. It's going to stop in the midbrain. If it's going to stop in the midbrain, we're not going up to the cortex. We're not going to the thalamus, so it's probably only going to have 2 orders of neurons, too. And then the spinal reticular. The reticular system, reticular activating system, or reticular formation is just in the brainstem. So we know it's going to the reticular formation somewhere in the brainstem. Again, it's going to stop in the brainstem. And since it's not going to really some of your brainstem is essentially conscious. You're consciously aware of this deep aching pain, but it's not going to be like you can. This is this deep aching pain. It's hard to localize. You just kind of like my whole back or body hurts or something like that, or my whole limb hurts. It's not like it hurts right here. Okay, so let's talk spinothalamic receptors, free nerve endings. Remember, all pain receptors and thermoreceptors always are free nerve endings. You just have to stimulate them to a certain level to get them to cause pain. They have a high threshold. You stimulate them enough, they cause pain. Spinothalamic is this fast, localized pain and temperature. So let's start with the leg. Let's start with the lower body. Here it is, foot. Follow my cursor, comes in. We have the cell body for the first order neuron is in the dorsal root ganglion, just like it was for conscious proprioception and every other. I don't know why my cursor keeps disappearing too. Pen won't wake up. So there it is. The type of axon that carries that's going to be either, most commonly, it's going to be an A delta fiber. Fast pain, A delta. Okay, that's an A delta fiber. Enters the lateral division of the dorsal root. So now pain and temperature comes in a little more, it comes in that dorsal root but a little more laterally versus all the proprioceptive stuff came in more medial. It comes in more laterally because it's going to actually go and make an immediate connection in the dorsal horn. So the cell body for the first order neuron is the dorsal root ganglion. And then we're going to get to the cell body for the second order neuron is going to be in what's called the first synapse is what's called the substantia gelatinosa, which is a region in that gray matter in that dorsal horn that typically involves mostly lamina 2, also lamina 1 and 5, but that's where it's going to make its connection with its second order neuron, which is going to then, let's just show you some of this stuff here. And again, I can't draw, so look at the red line here, the red structure. There's a cell body for pain and temperature. Dorsal root ganglion comes in the lateral division, more laterally out this way, of the dorsal root. Goes in here and makes a synapse somewhere in the substantia gelatinosa, which is this region right here. So you see substantia gelatinosa, which happens to actually correspond to this lamina, mainly lamina 2, but also some of 1 and some of 5, but mostly lamina 1 and 2 here. Okay, that region's called, it's like a nucleus, but it's called the substantia gelatinosa. Okay, it's a differentiated region of gray matter in that dorsal horn that has the cell bodies, the second order neuron cell bodies for fast pain and temperature. That axon from that cell body projects across this little anterior white commissure, just like our posterior spinocerebellar tract used that anterior white commissure. These guys use that same anterior white commissure, which is in this whole region right up here is called the anterior funiculus. But that anterior white commissure crosses over, and it enters into this region of the spinal cord called the spinothalamic tract. You notice it lies anterior and lateral. All these tracts we're going to talk about end up right there in that anterolateral region of the spinal cord. That's why it's called the anterolateral system. The spinothalamic tract is just makes up the biggest part of that anterolateral region. Okay, and then it ascends. But notice that it crosses essentially immediately. So it comes in at that spinal cord level and it crosses. What it does, it actually may go up or down a few segments before it crosses over, but essentially it crosses over very close to where it came in. It may go up and down a few segments. There's actually a little bit of white matter back behind, right in this region, right behind the dorsal horn, that it may go, the axon, as it comes in, it may go up or down a level or two before it makes its synapse. But essentially it does mostly crosses over right at the location that it. So then the second order neurons are in that lamina 1, 2, and 5, the substantia gelatinosa. Okay. They cross the anterior white commissure. That's the decussation, or the crossing point. They ascend. Then once they ascend here, they start out of the spinothalamic tract in the spinal cord. So here it is going up the spinothalamic tract. But once you get into the brainstem, it changes names. And it's called the spinal lemniscus. Kind of like the medial lemniscus, this is called the spinal lemniscus, coming from the spinal thalamic tract. So it's called the spinal lemniscus. Once it gets up all the rest of the way through the brainstem, it's called the spinal lemniscus. And if you notice, when we get up into the midbrain up there, it joins all the way up with the medial lemniscus, the trigeminal lemniscus, and the spinal lemniscus are all right next to each other. They have all eventually converge as they're getting close and getting ready to go into the internal capsule. They all are getting really close to each other, and then they go through the internal capsule together, okay? And then it goes, just like the conscious proprioception did, it goes, and it's from the body, it goes to the VPL of the thalamus. The axon goes up to the ventroposterolateral part of the thalamus. Anything that's conscious from the body is going to that VPL, okay, from the neck down, you know. So whether it's conscious proprioception or conscious pain and temperature, okay. And then it just goes then from then to the region. If it's lower body, it's going to come over here to this medial aspect of the homunculus and tell you about pain and temperature from that part of your body in the postcentral gyrus. I can't draw it out. We'll do that when we come back on Friday, likely. Now, let's mention the spinal limb. So, any questions on that's the spinal thalamic tract? Conscious. You're aware of pretty much pain at some type of conscious level. This is that one we just talked about is the fast localized pain. Bam, whack my thumb. Oh my God, it hurts right there. You know it hurts, okay? And it hurts right there on my thumb. After a short delay, you'll notice that you start to get this kind of pounding, deeper ache that may start to spread and just feels like your whole hand hurts, and it's this deeper aching pain. It's hard to say exactly where the pain is coming from, exactly where that hammer hit. It's the whole thumb now is irritated. That's what we're going to talk about now. These are often referred to as the divergent pain pathways because the information diverges. Once it gets in, it's going to different regions of your brainstem, okay, and to your cerebrum that are different than that localized specific part of that tell you exactly where the pain is. This is going to these other regions that help to then modulate pain. They're going to help to give you your psycho-emotional response to the pain, but also they're going to activate descending pathways that go to help modulate your pain and try to ramp it down, hopefully, a little bit. So that's what these guys are going to do. They're going to go up and they're going to stop in different locations and tell your brainstem to send signals down to your spinal cord to inhibit the pain. Or they're going to go up to your limbic system and say, oh, my mom always said just to toughen up and shake it off, and so I'm just going to shake it off. Or my mom always put a Band-Aid on everything and gave me all this attention, so I need to go seek some attention for my pain. When it goes to the limbic system, I need to be pampered and babied for a while. So this is where these pain pathways are going, this more divergent, slow aching pain pathways. Receptors are the same, free nerve endings. The modalities is slow aching pain that's difficult to localize. So when this is showing you kind of a close-up here. Let me bring my cursor down here. A close-up of the dorsal root. So when you're A delta and C fibers, this is mainly C fibers. Sorry, I should say C fibers, and that's what it shows there. This is so initially you get the fast pain. Boom, A deltas, send it up. Ooh, take my hand off the hot stove, move it, and then, ooh, that's starting to ache now. Now the C fibers that were delayed because they're a little slower, they start to seep in. The fast pain gets you to remove your system from danger, that the slow aching pain says, hey, something happened, it traumatized, it's still vulnerable, and it causes that aching pain, but it's on that slower C fiber, peripheral nerve C fiber. Enters the lateral division of the dorsal root, just the same as the other ones. It may ascend or descend a few levels in this thing called the dorsal lateral tract of Lissauer. That's this little white matter area right behind the dorsal horn that I mentioned, that it may go up or down a few levels before it makes its connection. It's going to go into the substantia gelatinosa, just like the other ones. It's going to happen, though, instead of using, other types of neurotransmitters, it's going to use substance P as its neurotransmitter, okay? Versus like a glutamate or something like that. which is a G protein neuromodulator, a little slower acting, a little longer acting. So it's using both C fibers that are a little slower in the transmission, but it's also using G protein mediated, slower acting um, neurotransmitter um, to then create the action potential, and then it all crosses over in the anterior Y commissure. So far, this is all matching up with the spinothalamic tract. And all these other divergent pathways all start out in the same location with the spinothalamic tract, and they all go into that anterolateral region, spinothalamic tract region up here, and then all go up together. And it's just a matter of where they end, okay? So you can see this is the spinal limbic. The spinal limbic is the one, I had to add it in here. It's this one right here. It's a blue one. Is that blue? Yeah, the blue. It goes all the way up to the thalamus and then goes to limbic system structures, okay? So the first order neurons are the same exactly as the spinothalamic tract, just that they use substance P as their neurotransmitter and using the C fibers from the periphery. The second order neurons are the same as well as the spinothalamic tract. Crosses in that AWC, the anterior commissure, ascends in the lateral brainstem as part of the spinal lemniscus. However, this is where it's a little different. Instead of synapsing in the ventroposterolateral part of the nucleus, it synapses in what's called the intralaminar nucleus of the thalamus, a little more deeper in the middle of the thalamus. And the way I remember that is the limbic system's kind of like in the middle heart of your brain. So the interlaminar nucleuses are kind of more in the middle of your thalamus. So they're kind of internal, interlaminar. So that's how I remember that the spinal limbic system, it's going to that middle part of my deep emotional part of my brain, is going to that more that deeper part of the thalamus and the interlaminar nucleus. So it doesn't synapse in the VPL, it synapses in the interlaminar nucleus. And then the third order neurons go from the interlaminar nucleus of the thalamus up through the posterior limb of the internal capsule again, just like everything else does, and then terminates in multiple areas of the cortex, including related to emotion and personality. Once it gets into the cortex, it goes to like your cingulate gyrus, it might go to your amygdala, it might go to your fornix, it goes to all these limbic system structures to tell you about pain as it relates to previous emotional responses and experiences and so forth. We're getting there. Spinomesencephalic tract. Spinomesencephalic. We're stopping at the midbrain. So here's what you need to know is everything else, it's all the same as the spinal limbic pathway, but the first part of the neuron here, it has two orders of neurons. It just has, since we're not going up anywhere to cortical structures, we're stopping in the brainstem, we'll only have two orders of neurons. It comes in the same dorsal horn, all that stuff matches up, crosses over in the anterior right commissure, goes up in the spinal thalamic tract, becomes part of the spinal lemniscus, but it goes up and it stops. This is it right here coming up to just the midbrain, okay? It carries information to two regions in the midbrain, to the superior colliculus, which we're actually, this cross-section through the midbrain is at the inferior colliculus, but it carries the little red one there is going to the periaqueductal gray. There's a little bit of gray matter right around the cerebral aqueduct that's going through the midbrain. And then the superior colliculus is just a little bit above that. A branch of it goes to the superior colliculus. What the spinal mesencephalic tract does is the superior colliculus, if you remember, is a visual relay reflex center that when you whack your hand, some of that information goes to that superior colliculus that makes you look. Before it even gets to your conscious awareness, your eyes are already turning to look for that danger. What caused that pain, okay? The other thing that signal does going to that periaqueductal gray, there's cell bodies in there that send axons down your spinal cord to go squirt a little dopamine and serotonin out at that level to start inhibiting the pain, to modulate the pain. So it's like your center for modulation of pain and resetting of the pain. So like, hey, how bad is this thing? Is it something that's not so bad that let's start toning the pain down a little bit? The initial stimulus was strong. We got out of danger. I'm feeling some aching pain. How bad is this? there's a delay there, but then your cerebral, then you have these descending pain pathways that are initiated by this spinal mesencephalic tract and also the spinal reticular pathway, which is next, that sends signals back down to tell your dorsal horn, hey, quiet down. Let's modulate the pain. Let's dampen, let's inhibit the pain a little bit now because it's the danger's over. The tissue's going to, be okay, that kind of stuff. So spinal reticular, it goes along the same initial, it's got two orders of neurons. This is reticular. There's a whole region throughout your brainstem that runs up and down through your brainstem, kind of in the middle meaty part of your brainstem. So kind of like this region here, that's kind of, you can see it kind of like freckled there. It's like dotted right there. That's the reticular formation. It's this big column of cells, and we're going to talk, you're going to hear this reticular formation come up a lot. There's a lot of things in that reticular formation that happen. So it's this area here, this area here, this area here. And you can see the little branches that are coming off. That's, again, divergent pathway. So that spinal reticular tract is just dumping off and sending axon projections to various levels of your reticular formation in the medulla, in the pons, and the midbrain. Those also activate regions in that reticular formation that send signals down your spinal cord to modulate pain. Okay? Some of the reticular formation fibers also project upwards into your cerebrum to help modulate pain, but mainly downwards to the spinal cord. Don't have to worry about the downward part yet. We're going to talk about that when we talk about pain modulation, and so forth in the next lecture. Okay? Spinal reticular. It's afferent, spinal cord, runs with all those anterolateral system pathways. It just stops at the brainstem. It only has two orders of neurons. All of these spinal, thalamic, anterolateral system, all of them are contralateral. So that's nice. They all cross, right? Everything's crossed except for spinocerebellar pathways. So everything's contralateral. Your sensory pathways are all contralateral, but your spinocerebellar pathways are all ipsilateral. That's kind of a general rule that you can kind of remember. So this, again, we start grouping things together as opposed to trying to memorize the individual details of every one of these. It's fine to try to do that once, but then you start looking for commonalities of these, and I'll try to help bring those out for you guys a little bit too. We're going to stop there because we're done. We don't have any more time. We actually got through a lot today, so thanks for hanging in there. That's because normally I stop like after the dorsal column, medial, and meniscal system for the first day. So you guys, we did it all except for the trigeminal thalamic in one day. So just go, just let it kind of sink in, let it, whatever, saturate your mind. So I will email you later to confirm that we do either do or do